Vegan diets do appear to help some people, at least in the short term. One key feature that both the vegan diet and the carnivore diet share is the elimination of certain foods, and it is this elimination which can be incredibly powerful. The mistake and judgment that is often made with regard to plant-based diets, however, is that it is the elimination of animal meat that produces the positive changes. Thousands of stories of those finding vastly improved health on strict carnivore diets or paleo and ketogenic diets that include meat argue strongly against this supposition. In an overzealous attempt to demonize animal foods, many overlook the fact that a vegan diet often excludes mainstream dairy and processed foods. We can't go blaming meat for the damage that the bread, sugar, and junk food have done. Today I will be reading to you my highlights of Dr. Paul Saladino's The Carnivore Code, Unlocking the Secrets to Optimal Health by Returning to Our Ancestral Diet. We know that most plants, including the vegetables beloved by all the diet experts, contain anti-nutrients that are difficult to digest and even toxic. That's why we must elaborately soak, sprout, ferment, and cook our plant foods to render them edible. Even then, many of us experience side effects like gas, bloating, and digestive pain that are so commonplace we have to come to view them as normal. As Paul will detail in this book, it's not normal to suffer when consuming meals that are supposed to be healthy. Carnivore seems extreme at first glance, but researchers confirm that human evolution was driven by a nutrient-dense diet, where calories came predominantly from nose-to-tail animals and secondarily from the plant kingdom. We most certainly evolved as omnivores. Don't let any whole food, plant-based zealot or carnivore stalwart tell you differently. But when Dr. Paul proposes that plants were merely the survival foods of evolution, we must carefully consider this premise. After all, as you will learn in this book, our ancestors' increasing consumption of nutrient-rich animal foods was strongly correlated with an increase in brain size. This was the primary catalyst for us to branch away from our leaf-chewing ape cousins and ascend to the top of the food chain. If you were going to construct the ideal diet for humans, what sort of metrics would you use to define this selection of foods? I would want this diet to include 1. All of the nutrients that we need to function optimally, 2. In the most bioavailable forms, and 3. With the smallest amount of toxins. I believe that a nose-to-tail carnivore diet is the best solution to it. But don't just take my word for it. That's what this book is all about. It's my shot at convincing you that animal foods are the best foods on the planet, and that plant foods are suboptimal, containing small amounts of nutrients that are much less bioavailable, and a myriad of toxins that do nothing but harm us. So now I ask you, do you want to be radically healthy? Do you want to have endless energy, mental clarity, a rocking libido, a sexy physique, and emotional resilience? Of course you do. We all want the best versions of ourselves and show up as powerfully as we can. This book is the story of how we can all reach our radical potential. These goals are attainable for every single one of us, and I believe that the biggest factor in achieving them is what we consume in our diet. The food we eat is the key determinant in whether we take the path toward obesity, brain fog, and fatigue, or the path towards optimization. As a whole, our health today as humans is pretty abysmal, and it's not getting any better. Estimates are that an overwhelming 87.8% of Western populations have some degree of insulin resistance and metabolic dysfunction. All I will say about the current medical system is that it will never be able to treat the root cause of illness until the fundamental paradigm of treating disease changes. Physicians need to realize and accept that food has everything to do with whether we become deeply sick or vitally healthy. Even though my parents were healthcare professionals, there wasn't much emphasis on healthy eating in our home. We ate a pretty standard American diet that included TV dinners, fast food, bread, pasta, and processed carbohydrates. I also grew up in a time period when fat was the enemy, having been wrongly demonized by the cereal and processed food industries since the 1950s. After school, I remember ravenously devouring multiple bowls of cereal, never feeling full. I also experienced irritability, childhood obesity, asthma, and eczema. I was a child of the low-fat era, and it showed. My greatest disappointment was in the overall medical paradigm and the medical system itself. Neither in the hospital nor in the clinic were patients getting better, and the progression toward worsening disease was constant. At times, their decline was slowed by medications, but the march toward morbidity was relentless. Suddenly, I had a paradigm-shifting thought that changed the course of my life from that moment forward. What if my own autoimmune issues and so many of the inflammatory problems we see manifested as chronic disease today could be triggered by the plants we are eating? Immediately, I dismissed the idea, burying it beneath a mountain of decades of indoctrination that plants, fiber, and phytonutrients were essential for human health. How would I poop without fiber? What about all the benefits of these so-called polyphenolic compounds? What about my microbiome? Wouldn't all the good bugs living inside my gut starve without prebiotic starches? It was the removal of plants that had resulted in this profound change in my experience of life, and I was deeply intrigued at what other benefits a carnivore diet might have. Since then, I have been eating only animal foods and am thriving like never before. My outlook on life remains extremely positive, my emotions are stable, my sleep is restful, and my body is strong. My energy is full, my libido robust, and yes, I poop every day and it is beautiful. Blood work, you ask? Remember that I am a physician obsessed with understanding how all of these things work. I've literally done hundreds of assays on myself, all of which have looked great. My kidneys and liver are healthy, and I definitely don't have scurvy. Nor do I have indications of inflammation or insulin resistance. In fact, my inflammatory markers are nearly undetectable, and my blood sugars are in the ideal range throughout the day without any significant change after I eat. What about my autoimmune disease? Since going carnivore, I haven't had a single eczema flare. Prior to making this change, I had been suffering from eczema every month, with many periods of persistent rash and itching. The majority of the disease and illness we experience today is autoimmune and inflammatory in nature, and I believe that by focusing on nutrient-rich animal foods while avoiding the toxins found in plants that trigger these processes, we will swiftly return to our ancestral birthright of radical health and vitality. We will explore why our decision to increase the consumption of plants was a bad idea. Ultimately, neither plants nor animals want to get eaten. But while animals have their legs, fins, teeth, and horns as defense mechanisms, plants are stuck in the ground and have been for 450 million years. During this time, in order to survive, they had to evolve complex chemical defense mechanisms that can wreak havoc on our bodies if we don't pay attention to them. It's okay not to want to be a carnivore or even carnivore-ish all of the time. As long as we are solving for the highest quality of life as much as possible, we'll be living amazing lives, full of rich experiences, personal growth, and profound health. The quality of life equation isn't meant to be a get-out-of-jail-free card or an excuse to eat cake. It's a gentle nudge to always be aware of what nourishes your soul most in the moment. It gives you the freedom to pause any effort to change your lifestyle if your highest quality of life temporarily changes. Our beginnings. Eating animals has been an integral part of our existence as humans and pre-humans for a very long time, probably for at least 5 to 6 million years. Primate evolution preceded ours by about 60 million years, and during that time period, the size of the primate brain stayed essentially constant at around 350 cubic centimeters, with some variation among species depending on body size. This research means that 60 million years of eating fruit and leaves didn't result in a bigger brain for our primate predecessors. It is thought that the hominid lineage diverged from chimpanzees about 6 million years ago when our distant ancestors came down from the trees and entered the open grasslands of northeastern Africa after the shifting of tectonic plates caused changes to their environment. The size of our predecessors' brains gradually increased after Lucy's time, and then about 2 million years ago, something incredible happened. They suddenly began to grow much more rapidly. They continued to increase in size, attaining an apex volume of 1600 cc about 40,000 years ago. This increase in brain size correlated with a growing complexity in the neocortex, the outer portion of our brains, and upgraded intelligence, both of which allowed for better communication and more sophisticated group behaviors, like organized hunting. Bigger brains equaled smarter people, and smarter people figured out how to hunt animals in groups more successfully. When we could hunt animals in organized groups with stone tools, we suddenly had first dibs on all parts of our kill. That meant we had access to the visceral, abdominal organs, and fat, as well as the muscle meat. I believe it was the eating of these parts of the animal, with all of their unique micronutrients and caloric abundance, that allowed our brains to grow beyond the initial increase in Homo habilis and made us into the humans we are today. Eating animals nose to tail is what made us human. Transitioning from scavengers to hunters appears to have been the defining moment in our evolution as humans. Some would argue that it was cooking that resulted in the sudden increase in the size of the human brain, but many scientists agree that our torrid love affair with fire did not begin until about 500,000 years ago, 1.5 million years after our brains began to grow exponentially. Extremely high levels of delta-15N isotopes suggest that 40,000 years ago, Homo sapiens and concurrent Neanderthals were high-level trophic carnivores. They were consuming the vast majority of their protein from large mammals like mammoths rather than from plant sources. From an energy efficiency standpoint, hunting large animals makes more sense. Gathering plants and stalking small animals deliver much less of a caloric and nutrient bounty relative to the energy that is invested. Across more recently studied indigenous peoples, we observe a similar pattern that clearly indicates preference for animals over plant food. But our ancestors weren't just looking for any animals. They were looking for the animals with the most fat on them. Larger animals have more fat by weight, and fat appears to be the macronutrient we sought most for survival. There are many sources of protein in the animal world, but the sources of fat are more scarce. By weight, fat provides more than twice as many calories than do protein or carbohydrates. In addition, the human metabolism makes fat a uniquely valuable and necessary food. If we think of ourselves as automobiles that need fuel for our metabolic engines, we shouldn't put protein into our gas tank. For best results, our metabolic engine runs more efficiently on fat or carbohydrates. Historical accounts of Arctic explorers tell us of the dangers of rabid starvation, a condition that occurs when we eat too much lean protein without carbohydrates or fat. Our liver has a limited capacity to turn the nitrogen from amino acids into urea, a water-soluble compound that we excrete in our urine. The upper limit for protein in our diets seems to be about 40% of our total calorie intake, beyond which point we might exceed the liver's ability to process this macronutrient. This means that 60% of our caloric needs must be met by either fat or carbohydrates. Aside from occasional fruit and tubers, the stems and leaves of plants don't contain much in the way of carbohydrates, and they are often riddled with plant defense chemicals as well. The vast array of carbohydrate-rich plant foods we see in grocery stores today is nothing like the selection of plants in the wilderness or the plants our ancestors would have had to choose from. I want to share with you my Carnivore Code hypothesis. I believe that throughout our evolution, our ancestors have hunted animals preferentially and have eaten plant foods only during times of scarcity or starvation. I base my hypothesis on the following factors. 1. Anthropological data including brain size, stable isotope data from bones and teeth, and examples from indigenous peoples as we have just discussed. 2. The significantly higher availability of energy from animals relative to plant foods per energy invested. 3. The vastly superior nutrient content of animal foods. Critics of the carnivore diet love to point out that our teeth don't look like the teeth of other carnivores like lions or tigers. However, this isn't really a fair comparison because our evolutionary lineage is completely separate from felids. Depending on the availability of food sources, our mouths appear to be adapted to eating both plant and animal foods. As we move down the digestive tract, however, we begin to see stronger inclinations towards eating animal foods. The pH of a healthy human stomach is around 1.5, which is very acidic. Our distant primate ancestors have a stomach pH of 4 to 5, which is much less acidic. pH is a logarithmic scale, and every increase of 1 translates to a 10 times less acidic solution. Our stomachs are about 1,000 times more acidic than a chimpanzee's. 1,000 times is no joke, and it certainly wasn't an accident. Our stomach became much more acidic because 3 to 4 million years ago, our diets changed from predominantly plant foods to including many animal foods, and then 2 million years ago to mostly animal foods. The low pH in our stomach protects us from pathogens in the environment and breaks down foods so intensely that it's not seen as foreign by the immune system that resides in the walls of our intestinal tract. Pharmaceutical medications like proton pump inhibitors raise pH levels and thereby increase the risk of contracting pneumonia, infections, and numerous allergies. Maintaining a low stomach pH was, and continues to be, critical for optimal human health. The fact that it's much lower than our primate ancestors is no accident. It points directly to a radical change in diet early in our evolution as hominids and indicates selective pressures for those best adapted to eating fresh and not so fresh animal meats. In primates, plant matter hangs out in the massive large bowel, where it undergoes fermentation and it results in the production of large amounts of short-chain fatty acids, which are then used as the main source of their calories. Even though primates are eating mountains of carbohydrate-based plant matter, they are actually running on fat. They need such large colons to house the bacteria that ferment all of the plant fiber they eat into the fats for energy. If you've seen monkeys or apes, you'll notice that their rib cage angles outward with protuberant bellies to accommodate their commodious colons. Both brain and intestine are very metabolically active tissues that require a lot of energy to function properly relative to their mass. A gram of brain tissue requires 22 times the amount of energy to function as a gram of muscle tissue. Our intestines are similarly greedy. As the gut shrank in size and in energy needs, the brain was free to gradually expand over generations into the stellar instrument we possess today. With shrinking guts also came straighter rib cages and flatter bellies. Members of the Homo heidelbergensis species that stayed in Africa appear to have evolved into our direct predecessors, Homo sapiens, who left Africa much later, around 70,000 years ago. We don't know why our more recent Homo sapien ancestors left Africa at this time. For the majority of our evolution as humans, we were probably not eating many starchy foods like tubers, and the consumption of these types of foods is likely a recent adaptation due to the shifting of food availability. Many have hypothesized that our migration out of Africa 80,000 years ago was related to declining populations of megafaunal animals due to overhunting. DHA doesn't occur in plants, and humans are horrible at converting the precursor form of omega-3 into this precious brain-building block. Relative to other animals, humans aren't great at sprinting, but we are very good at the walking and running of long distances that would have been needed to track down and hunt animals. Our feet also changed during this time to assist in long-distance running and walking. The big toe became more aligned with the rest of the foot, and our heel pad grew to provide cushion as we walked more upright. There were also changes in our knees and spine to optimize posture and upright mobility. Our ancestors realized they were better off cooperating in endeavors like hunting or fighting off attackers. In these situations, knowing where our compatriots were looking was a very good thing. The whites of our eyes are thus believed to have been one of the key adaptations that evolved over the last 6 million years and that allowed us to communicate with tribe members in silence. By showing them where our attention was directed without speaking aloud, we could communicate more quickly and with greater stealth. Lions and tigers are well adapted to chasing down a gazelle or a wildebeest on the open plains, but they are specialized for only that type of prey. On the other hand, over the last 5 million years, humans have evolved to acquire elegant shoulder joints, upright postures, and pelvic girdles that have allowed more fluid running, as well as more acidic stomachs, smaller guts, white sclera, and bigger brains. These adaptations have all propelled our species toward one destiny, becoming the best hunters this planet has ever seen. Our worst mistake. It is only when we suddenly lose our health that its value becomes clear. When that happens, we recognize that nothing matters more than regaining and recovering our vitality. It is only then that we will be able to resume our lives and enjoy the natural world around us as we care for our families and share our time with others. What if I were to tell you that at one point in our evolutionary history, our health went from appearing pretty good to being basically abysmal almost overnight? Well, this is exactly what happened about 12,000 years ago during the Neolithic Revolution, when we began farming and joined the cult of the seed. Although we live longer today than at any other point in our past, our overall health is inferior, and chronic disease is rampant despite the mountain of technology and other advancements we've created. Claiming that the advent of agriculture is responsible for many of the pleasantries we enjoy today is an overly bold intellectual leap, reliant on many unexamined assumptions. During the first 15 years of their lives, they are 75 to 199 times more likely to die than individuals in Western societies. That is, from birth to age 15, you'd be much more likely to die as a hunter-gatherer than a Westerner. The factors that contribute to this mortality hazard include less access to clean water, lack of sanitary waste facilities, infectious illnesses, and traumatic wounds. Comparisons of life expectancy between these groups are confounded by these inflated childhood mortality rates. When we compare the health, vitality, and quality of life in elderly indigenous groups to that of Westerners, the former are clearly superior in all aspects. Studies of present-day hunter-gatherer tribes invariably reveal robust health and freedom from chronic disease like diabetes, depression, and dementia. Many studies across varied populations show a strong correlation between adult height and nutritional quality. One study looking at male height across 105 countries came to the following conclusion. In taller nations, the consumption of plant proteins markedly decreases at the expense of animal proteins, especially those from dairy. Their highest consumption rates can be found in Northern and Central Europe, with a global peak of male height in the Netherlands, 184 centimeters. Like the Indian, the Eskimo thrived as long as he was not blighted by the touch of modern civilization, but with it, like all primates, he withers and dies. In his primitive state, he has provided an example of physical excellence and dental perfection, such has seldom been excelled by any race in the past or present. Based on energetic and nutrient superiority, our ancestors have always favored animal foods over plant foods when animal foods were available. Current estimates of the plant-to-animal consumption ratio in present-day hunter-gatherers suggest an approximate 50-50 split. But as many have pointed out, these populations no longer have access to large game and are forced to adapt to their changing environment by gathering more plant foods. Thus, currently living populations of hunter-gatherers are a poor indication of the relative amounts of animal and plant foods consumed by more distant predecessors. Chemical warfare. Welcome to the world of plants. When insects and animals decide to go munching on our green neighbors, plants can't run away, bite, or verbally threaten them to defend themselves. Ever go hiking in the desert and accidentally bump into a cactus? Ever play in the woods and stumble into a patch of brambles? Ouch and double ouch. We are much less aware of the molecular mechanisms plants have evolved to defend themselves, however, and these chemical spikes can cause just as much harm as the spines on a cactus or the thorns on a rosebush. Most of the compounds we think of as phytonutrients are in fact phytoweapons, meticulously designed by plants to discourage insects, animals, and fungi from consuming them for breakfast. Plants do not want to be eaten. Sure, fruit is a different story, and we'll talk about that later, but it's still not that good for you. Animals don't want to be eaten either, and I couldn't agree with this more. However, remember that animals have evolved different sorts of defense mechanisms to combat predators. Usually, this is simply getting the heck out of Dodge when a hungry predator shows up. There are literally hundreds of thousands of chemicals produced in the plant kingdom that can harm us. Broadly speaking, we can divide these toxic compounds into a few categories. The largest category is the phytoalexins, chemicals directly produced by plants to ward off attack from insects, fungi, and animals. This category includes many of the compounds that we have been told are good for us, including glycosinates and many polyphenols. The most well-known polyphenols are probably resveratrol and curcumin, And if you believe the hype, by taking these you'll live forever, cleanse yourself of inflammation, and maybe even grow wings. In his comprehensive scientific article titled Dietary Pesticides, 99.9% all natural, researcher Bruce Ames highlights the fact that humans consume 99.99% of their pesticide load from plants themselves and only 0.1% as pesticides sprayed on food. Glyphosate and other synthetic pesticides are certainly harming people, but the amount of these consumed pales in comparison to the quantity of plant-produced pesticides in our diet. The human intake of these toxins varies markedly with diet and would be higher in vegetarians. Our estimate of 1.5 grams of natural pesticides per person per day is based on the content of toxins in the major plant foods. Plant pesticides are pervasive, with at least 42 compounds known to occur in a seemingly benign food such as cabbage. Furthermore, many of these compounds have been shown to be damaging to DNA in humans and animals, a process known as clastogenesis. Plant molecules like phytolexins, polyphenols, oxalates, and lectins are like computer programs from a different operating system than ours. So-called antioxidants from plants don't directly serve an antioxidant role in our body. In fact, they are often doing the opposite, acting as pro-oxidants. Our body has its own intrinsic antioxidants, like glutathione, that are part of our programming and that manage the balance of oxidation and reduction just fine on their own. In fact, multiple studies have shown that supplementation with plant antioxidant molecules do nothing to improve antioxidant status in humans and are often associated with worse outcomes. The plant-based forms of many vitamins and nutrients look very differently from the corresponding animal-based forms. Beta-carotene versus retinol, vitamin A, alpha-linoleic acid, ALA, versus DHA, omega-3 fatty acids, and vitamins K1 versus K2 are all examples of this. We'll also discuss the many nutrients key to optimal human function that don't occur in appreciable amounts in plants or fungi, like vitamin B12, choline, taurine, carnitine, carnosine, vitamin K2, and others. At a biochemical level, life can be distilled to the exchange of electrons between molecules and the harvesting of energy stored in bonds between atoms. We eat food in order to gather energy stored in the bonds of its molecules, translating this energy through the movement of electrons into other storage forms of energy, namely into a molecule called ATP, or adenosine triphosphate. Oxidation and reduction refer to the molecular loss and gain of electrons, respectively. When a molecule is oxidized, it loses an electron to another molecule, which is simultaneously reduced as it gains that electron. Free radicals are molecules with an unpaired electron, and they are an unruly sort of character. They are highly reactive and have a predilection for stealing electrons from proteins, lipids, and nucleic acids, which results in oxidation of these molecules. Free radicals aren't entirely bad, however, and they do serve important signaling roles in the human body, but if overproduced, they can cause oxidative stress, a condition in which the delicate balance of oxidation and reduction is thrown off, resulting in cellular damage and aging. While humans have been feasting primarily on animal foods for the last 2 million years, similar selective pressures from plant toxins have not been present for us, and we appear to be much less adapted to eating significant amounts of plants for long periods of time. Moose and many other grazing animals have evolved proteins in their saliva to deactivate tannins present in leaves, which act as digestive enzyme inhibitors. Ruminants also possess multiple stomachs, which accomplish the task of digesting plants and breaking down plant toxins much differently than monogastric, single-stomach animals like humans do. Rabbits and other small animals often chew plants like sagebrush excessively, allowing for many of the volatile plant toxins to be off-gassed in the process. When populations of herbivorous animals like deer and buffalo are overcrowded and forced to abandon their innate feedings and grazing patterns, mass deaths occur as a result of overconsumption of only a small variety of plants. Broccoli. Superhero or supervillain? Broccoli belongs to the Brassica family of vegetables, all of which are derived from an ancient type of mustard plant. The many phases of this family, also known as crucifers, include kale, collard greens, Brussels sprouts, cabbage, kohlrabi, horseradish, wasabi, Swiss chard, cauliflower, rutabaga, bok choy, watercress, radishes, mustard, and turnip. A unique feature of this family is the presence of the sulfur-containing compounds known as glucosinolates, which are transformed into isothiocyanates and related compounds when acted on by the enzyme myrosinase. Sulforaphane does not exist in a healthy living broccoli plant. It only shows up as a defense chemical in response to damage being done to the plant cell walls. Sulforaphane is so toxic that it can't be present in a healthy broccoli plant, or it would cause massive damage due to its strong capacity as a pro-oxidant. How does sulforaphane do its dirty work? In animals, including humans, it has two main mechanisms of toxicity, a slow one and a fast one. The fast mechanism of harm is accomplished by acting as a vicious pro-oxidant, causing the formation of free radicals that damage the delicate lipids in cell membranes, proteins, and DNA. Sulforaphane is only one of 17 isothiocyanate compounds known to be present in broccoli. Other cruciferous vegetables have even more. Cabbage, for instance, demonstrates 42 known plant toxins. When we eat broccoli or any plant from the Brassica family, some of the absorbed sulforaphane is immediately detoxified because our body knows it's a toxin and does not want it. Whatever is not broken down circulates in the bloodstream and competes with iodine for absorption at the thyroid, preventing this gland from getting one of the minerals it needs to make thyroid hormones. Cases of hypothyroidism induced by overconsumption of crucifers have been reported even in westernized populations, but are extremely common in underdeveloped regions of the world. Cassava is a plant native to South America, the roots of which are widely consumed as a source of carbohydrates in poor regions of the continent where there is limited access to more nutrient-rich food. In all parts of the plant, but concentrated in the roots, is the molecule linamarin, which is a cyanogenic glycoside. These compounds are also found in the pits of stone fruits like peaches, apricots, and plums, and they are highly toxic. Linamarin itself isn't toxic, but when combined with the enzyme linamarase, it's broken down into hydrocyanic acid, which is exactly as toxic as it sounds. When ingested, hydrocyanic acid quickly breaks down into cyanide, an extremely potent mitochondrial poison. Cyanogenic glycosides in other foods, like the aforementioned pits of stone fruits, can similarly break down into cyanide, and poisonings have been reported with overconsumption. In order to be eaten, cassava must be either fermented for 3 days or ground up and left to dry in the sun, while the majority of the hydrocyanic acid is released into the air. Chemotherapy agents are vicious chemicals that kill both cancer and native cells, often leading cancer patients to the brink of death. Sulforaphane has been shown to have potential benefit in cancer-fighting models because it damages cells and can induce programmed cell death or apoptosis. But when introduced into our bodies, it's going to target our healthy cells too. Sulforaphane can create so many reactive oxidant species in both cancerous and non-cancerous cells that programmed cell death is initiated. Generally speaking, any type of molecule that causes oxidative stress in our body activates this pathway so we can ramp up our defenses. This includes tobacco smoke, heavy metals like lead, mercury, and arsenic, alcohol, oxidized vegetable oils, and hyperglycemia. While it is true that sulforaphane does act as a pro-oxidant by activating the NRF2 system, it induces the formation of our own endogenous antioxidants like glutathione. There are studies showing that administration of sulforaphane improves our antioxidant status in the short term, but this is related to increased production of glutathione, not to any direct antioxidant capacity of sulforaphane. If you still want to believe that broccoli is really healthy for you, then you can make it even healthier by drenching it in oxidized canola oil while smoking a cigarette and inhaling diesel exhaust. That sounds absurd, doesn't it? But all of these compounds turn the NRF2 pathway on, just like sulforaphane. The concept of xenohormesis assumes that we need the compounds found within vegetables to achieve optimal antioxidant status, and that eating crucifers somehow elevates our glutathione above baseline levels long term, thus making us superhuman. Sadly, these assumptions are not the case. There have been multiple interventional studies comparing diets rich in fruits and vegetables, including crucifers, to diets with low or zero amounts of these plant foods, and the results have shown zero benefit to eating such foods. We don't need fruits and vegetables to achieve robust antioxidant defenses, and there isn't any evidence that consuming these plant foods makes us healthier. As we discussed earlier in this chapter, however, there is significant evidence that these plant compounds are harmful, damaging DNA and interfering with hormonal signaling throughout the body. Of hormesis, it is often said that the dose makes the poison. I believe this is true with environmental hormetics, but not molecular hormetics like isothiocyanates, polyphenols, smoking, etc. Because of the often ignored side effects of these molecules on the body as a whole, they are toxic from the get-go, even in small doses. Environmental hormetics and molecular hormetics do not operate the same way and should not be confused. There are multiple examples of environmental hormetics: heat, cold, sunlight, exercise, and if we experience these in the setting of a nutrient-rich diet, they are all we need to obtain robust levels of antioxidant defense. Too much exposure to any of these will certainly be damaging to our body, but small amounts act as stressors that temporarily cause oxidative stress with glutathione depletion, followed shortly thereafter by a rebound to levels higher than baseline. Just because those eating more meat appear to have worse health, it does not mean that it was the meat that caused the problem. It's much more likely to have been the things most of the population eats with meat: refined grains, bread, sugars, and other junk foods. Epidemiology simply can't distinguish between all of those possibilities. It can only show correlations, but cannot describe causal connections. That's what interventional studies are for, and when we look at this type of research with meat, we find that it is clearly not bad for humans in ways that those misled by epidemiology might believe. Of unicorns and fairy tales. Perhaps more than any other plant molecule, polyphenols have become associated with the term superfoods. They are often dubbed antioxidants, and we are told to consume as much of them as possible. The term polyphenol refers to the molecular structure of a class of organic compounds. Poly means many, and phenol refers to the aromatic multiple double bonds between carbons ring structure with an OH group attached. In the plant kingdom, polyphenols serve unique roles as phytoalexins and as plant pigments. You'll recall from the last chapter that phytoalexins are weapons that plants marshal against attacking organisms like fungi, insects, or animals. Resveratrol, for instance, is produced as a defense molecule in the skin of grapes and other plants when they are attacked by pesky fungi. Intriguingly, but not surprisingly, humans and animals do not make molecules that are polyphenolic in structure within their biochemistry. Although there are many epidemiology studies that show some degree of correlation between consumption of fruits and vegetables containing polyphenols and improved health outcomes, many interventional studies don't reveal any iota of benefit. Most of the interventional research that is done within medicine today is funded by the pharmaceutical companies who stand to profit handsomely if their molecule du jour generates compelling data. If an interventional study shows that meat is good for you, cattle farmers aren't exactly going to get rich. During the 1930s and 1940s, meat, fat, and other animal foods were viewed as healthy dietary choices that will make you strong and vital. Then suddenly, around the 1950s, that narrative began to change dramatically. We were told that butter was bad for us, saturated fat from animals would give us heart attacks, and that we should be consuming more plant foods and vegetable oils. As a whole, this has been the overarching narrative since that time. Over the last 70 years, what type of people were eating lots of vegetables? It was those who were listening to mainstream nutritional advice trying to lead healthy lives. These people were also much more likely to have been practicing other types of healthy behaviors like meditation, stress reduction, paying attention to sleep, exercising, and avoiding junk food. Because fruits and vegetables aren't cheap, they were also much more likely to be consumed by those of higher socioeconomic status with access to good medical care. What if it were these healthy behaviors that resulted in better health outcomes rather than the fruits and vegetables suggested by the epidemiology studies? We see that in Asian population, consumption of animal foods was associated with better health outcomes, in this case with respect to stroke. Both of these studies are epidemiological, which always comes with limitations, but they and many other investigations paint a very different picture from those done in the West. This contrast calls into question the validity of the latter's findings and strongly suggests healthy user bias as a confounder. It was probably the healthy behaviors in both groups of people that led to improved mortality rates rather than the avoidance of meat and fish by the vegetarian group. If the investigators had only compared this vegetarian group to the general British population, it might have looked like it was their dietary choice that improved longevity, and this is exactly how healthy user bias can be misleading. Because meat has been associated with the potential for ill health in Western culture for the last 70 years, those people who are eating more meat are the rebels. These are the James Dean type who don't care what they are told. They are going to eat meat even if they are told it's bad for them. And they are also probably going to smoke, ride motorcycles, not exercise, and eat a bunch of junk food with their meat. We must not blame the meat for what the soda, bread, junk food, smoking, and a sedentary lifestyle have done. This is the unhealthy user bias at play. Damaging DNA is a very bad thing. Curcumin appears to do this, as well as harming other cellular structures, by creating reactive oxidant species, free radicals, that result in oxidative stress. See a pattern here? In a horrid case of mistaken identity, the molecules that are marketed as being antioxidants are, yet again, actually pro-oxidants. Curcumin's bioavailability is vanishingly small, and what is absorbed is quickly detoxified in the liver and excreted. The amount of this molecule our body absorbs from eating turmeric is likely going to be pretty small, and our body gets rid of it quickly to avoid toxicity. Contrary to popular belief, the liver does not store toxins, but instead functions as the epicenter of toxin modification and excretion. Liver is an incredibly nutritious food that belongs in any diet, and nothing like the filter is so often made out to be. Though inflammation isn't usually a good thing to have happening in our bodies, it's a valuable signal that things are out of balance. Rather than reflexively seeking to throw heaps of anti-inflammatory molecules at it, whether they be plant-derived or synthetic, we should first seek to understand where it's coming from. At a biological level, inflammation is activation of the immune system, which is incredibly complex and made up of many types of cells. When activated, these various cells signal to each other that something is awry with molecules called cytokines that travel throughout the body and cross the blood-brain barrier. I strongly believe that most, if not all, chronic disease we suffer from today is inflammatory and autoimmune in nature. Autoimmunity occurs when the immune system begins to see our own body as foreign and attacks it as it would an invader. The state of optimal health includes a balance between the processes of oxidation and reduction, not a complete abolition of these reactions in our body. Not surprisingly, many studies using antioxidant interventions reveal an increase in mortality outcomes. There are many things that can cause inflammation, but the food we put into our bodies is the greatest contributor of them all. Things like stress, inadequate sleep, and environmental toxins can contribute, but the impact of what we eat far outweighs these. When food causes inflammation, the main pathway by which it does so is by damaging the gut with the subsequent activation of our immune system army that resides on the other side of the delicate intestinal epithelial cell layer. Antigens from food aren't supposed to get across the gut lining, but when they do, they look like invaders and cause the immune system to go into high alert. What results is the loss of gastrointestinal barrier function as well as subsequent immune activation, which colloquially is known as leaky gut and appears to be at the root of the majority of the chronic illnesses that we experience today. So which foods damage the gastrointestinal epithelium and cause leaky gut with its potential for downstream kindling of the immune system? Which foods are full of toxins, chemical weapons, and anti-nutrients designed to do exactly this? You guessed it, plant foods. Do animal foods have similar sorts of toxins and weapons in them? Nope. Animals don't need these because they can flee or fight. Plant molecules do not act directly as antioxidants in the human body, nor would this sort of disruption of our redox balance be advantageous. The story of flavonoids is similar to that of sulforaphane. When these molecules demonstrate improvements in oxidative stress parameters, they do so through the NRF2 system by first acting as pro-oxidants. Quercetin, a molecule found in onions, berries, grapes, and peppers, is also known to trigger estrogenic signaling and act as an endocrine disruptor. It accomplishes this both by binding to the estrogen receptor and interfering with the breakdown of estrogen. Extracts from raw cabbage, fermented cabbage, and Brussels sprouts stimulate estrogenic signaling in cell culture breast cancer models. When a compound stimulates estrogen signaling in cell culture models, it's going to behave similarly in our bodies and will usually be damaging for both men and women. Many herbivorous ruminant animals like moose and sheep have developed proteins in their saliva that bind to tannins and inactivate them. Animals that have consistently evolved eating plants as their main source of food appear to have developed multiple mechanisms like this to help them deal with plant toxins, but humans are lacking in this department. Without such protection, these molecules inhibit our digestive enzymes when ingested and impair the digestive process. In order for DNA to be turned on and transcribed, portions of our chromosomes must be unwound to expose the DNA. Our DNA sequence constitutes our genetics, and the way genes are turned on and off through unwinding of chromosomes is known as epigenetics, a process controlled by the addition or subtraction of methyl groups and other molecules to histone proteins. We can't change our genes, but we absolutely can change the way they are turned on and off. How? Through diet and lifestyle. These are the most powerful tools we have to create health in our lives. Though salicylates are not technically polyphenols, these molecules do contain one aromatic ring and are used in plants as defense hormones in response to attack. Foods high in salicylates include asparagus, almonds, avocados, cherries, nectarines, dates, blackberries, coconuts and coconut oil, honey, tomatoes, potatoes, and eggplants. Just a side note here, Dr. Paul Saladino has since introduced honey into his diet and various other fruits. By taking resveratrol, they claim, you can become a supermodel vampire, forever young and vital. Sadly, these claims were made prematurely and based entirely on animal studies. To be fair, the results in worms, fruit flies, and mice were impressive. They showed improvements in models of diabetes and increased longevity. The mechanism of resveratrol, which appears to turn on a family of genes called sirtuins involved in longevity, is intriguing. Be that as it may, in human trials, resveratrol has been a massive failure and also shown many potential damaging side effects. My point here is not to say that activation of NRF2 is always a bad thing, but to highlight the following fact. Isothiocyanates, smoking, alcohol, exercise, heat, cold, and polyphenolic molecules are pro-oxidants rather than antioxidants. As we touched on previously, molecular pro-oxidants, even at small doses, possess damaging side effects like endocrine disruption and inhibition of detoxification systems, whereas the environmental inputs do not. This is the key difference between hormetic plant compounds and environmental hormetics. Another interesting wrinkle to the resveratrol story is that we don't need this molecule to activate the sirtuins, which potentially improve longevity. Guess what else activates these genes? Ketosis. Ketones like beta-hydroxybutyrate turn on the sirtuins and many other genes associated with longevity. We don't need the empty promises of plant molecules to be optimal. Jumping in cold water, exercising, and fasting or eating a ketogenic diet like the carnivore diet allows us to achieve optimal health and avoid the negative side effects that come with plant molecules not compatible with our human operating system. Another side note here, Dr. Paul Saladino has since changed his position on the health effects of long-term ketogenic diets and talks about the harmful effects of ketogenic diets for the long term. Avoiding inflammation, turning on longevity genes, and achieving healthy hormonal balance are all things that are easily discovered with a nose-to-tail carnivore diet, and all of which can be attained without harmful plant compounds. Attack of the oxalates. We're going to talk about a molecule that is produced in small quantities as a waste product of human metabolism, but occurs in very large amounts in some plants and can cause serious pain, kidney stones, and other issues when it accumulates in our body. I speak of oxalates, the colloquial term for a combination of oxalic acid and a mineral such as calcium or magnesium. In general chemistry terms, when a molecule or an atom with a positive charge connects to another molecule or atom with a negative charge through an ionic bond, a salt is formed. Technically, calcium plus oxalic acid is a calcium oxalate salt, but for the purposes of this discussion, I'll use the term oxalate to refer to both oxalic acid and its salts. In humans, oxalic acid is produced as a waste product and excreted in the urine. It occurs as the result of the breakdown of the amino acids glycerine and hydroxyproline and of a molecule called glyoxylate, in a process termed the Glyoxylate Pathway. In a small number of individuals with a rare genetic disorder known as primary hyperoxaluria, or PH, mutations in this pathway lead to much larger endogenous formation and excretion of oxalates, 100 to 600 milligrams per day. People with primary hyperoxaluria suffer from the formation of frequent and severe calcium oxalate kidney stones, often leading to permanent kidney damage or renal failure, as well as oxalate deposition in bones, joints, bone marrow, and other tissues outside of the kidneys. This is a condition known as systemic oxalosis. In plant biochemistry, oxalic acid is actively formed during the process of photosynthesis and has many roles. For example, chelating or biting minerals such as calcium, magnesium, and zinc to regulate their concentrations within cells. Plants also use oxalic acid as a defense weapon against predators. Oxalic acid crystallizes into multiple shapes in both plants and humans. One of these is the rhaphite form. Some plants, widely considered to be healthy, contain huge amounts of oxalates relative to what we might naturally find in our bodies. Normally, humans produce 10 to 30 milligrams of oxalates per day through the glyoxylate pathway and amino acid breakdown. By simply downing one healthy green smoothie in the morning, we could be exposing ourselves to more than 200 times this amount. What do most people put in their green smoothies? A concoction of spinach, almond milk, and blackberries, with a scoop of turmeric thrown in for good measure, could easily create a drink with over 1,000 milligrams of highly absorbable oxalates. Is that a lot? You bet it is. There have been documented cases of deaths related to consumption of as little as 5,000 milligrams of oxalates, a dose that is attainable in one day from something like a green smoothie cleanse. Quote, diet-induced hyperoxaluria has been reported with starfruit, nuts, peanuts and almonds, rhubarb, iced tea, chaga mushrooms, juicing from fruits and vegetables, and vitamin C, unquote. Furthermore, in children with genitourinary problems ranging from bloody urine to kidney stones, removal of almond milk has been shown to resolve these issues. In healthy individuals, microscopic and macroscopic large oxalate crystals have been observed pathologically in the thymus, kidney, blood vessels, testicles, brain, eyes, thyroid, and breasts. Oxalates do not serve a purpose in these tissues, but appear to be deposited in them over our lifetime, which is likely connected with increased levels in the blood from consumption of high oxalate-containing foods. Over 75% of kidney stones are made from calcium oxalate. This means that around 3/4 of all kidney stones could possibly be prevented by avoiding oxalate-containing foods. By consuming an ordinary amount of only one food that is high in oxalate, we can create levels of oxalate in our bodies that are comparable to someone with primary hyperoxaluria. Of kidney beans and Parkinson's disease. Ricin is a poison derived from castor beans and is the most deadly lectin known. A dose the size of a few grains of salt can kill an adult human. During World War I, the US military considered coating bullets with ricin, and it has been used for many covert assassinations within the last 70 years. Whether it's inhaled from a letter in a powdered form or injected as a pellet by a devious secret police agent, ricin kills victims over the course of a few painful hours by inhibiting the function of ribosomes, cellular organelles where RNA fragments are made into proteins. It basically gums up our cellular factories, grinding them to a screeching halt and slowly ending the life of any organism unlucky enough to ingest it. Not surprisingly, castor beans do not make good food, and the consumption of 5 to 20 castor beans has proven fatal to humans. The concept of lectins can be a bit confusing, but simply put, they are a special type of protein that binds to glycoproteins on the surface of or within our cells. In the case of ricin, this lectin binds to a carbohydrate portion of the ribosome and prevents it from doing the crucial work of protein formation. Generally speaking, lectins tend to be the highest in the roots and seeds of plants, and foods with the highest content of these disruptive proteins are usually legumes, grains, seeds, nuts, and tubers. Though usually not as potent, many other plant lectins can also be toxic and have caused poisonings. Acute nausea, vomiting, and diarrhea have occurred in humans who have consumed undercooked kidney beans, which contain a lectin known as phytohemagglutinin, or PHA. There are hundreds of recorded food poisoning outbreaks related to this seemingly innocuous bean and its powerful lectin. Having a diverse array of microbes within our gastrointestinal microbiome is crucial for optimal health, and low diversity has been associated with a variety of chronic diseases, including diabetes and inflammatory bowel illnesses. PHA and many other plant lectins appear to be damaging to our gastrointestinal tract by decreasing the diversity of organisms there through interacting with our gastrointestinal epithelial cells. Everything we eat and the trillions of organisms that live inside of us throughout our life are separated from armies of our immune cells and the inside of our body by one cell layer. This is a very important layer of cells. This gastrointestinal epithelium has the crucial job of absorbing the nutrients we need to grow and thrive, while simultaneously keeping pathogens out. It is composed of a variety of unique cell types that all originate from progenitor stem cells found in the base of folds within the membrane of the small intestine. The main cells within the epithelium are known as enterocytes and possess tiny finger-like projections called microvilli, which exponentially increases the surface area of the small bowel. They are crucial for the proper absorption of nutrients from your food. In autoimmune conditions like celiac disease, regression of these microvilli is observed and the surface of the small intestine becomes pathologically smooth, causing malabsorption of nutrients and leading to profound nutritional deficiencies. It appears that PHA is binding directly to the gut lining and damaging the cells there, including the goblet cells that help create and maintain a healthy mucus layer. As this mucus layer is eroded by the actions of PHA, bacteria come into contact with the gut epithelium, which causes our body to sound the alarm to mount an immune response. In animals and humans, lectins like PHA appear to be causing harm by damaging the integrity of our gastrointestinal epithelia and creating leaky gut. A single layer of cells composing the gastrointestinal epithelium serves the invaluable role of keeping bad stuff out and bringing good stuff in. Specialized cells within it create the mucus layer that serve as a buffer between microorganisms inside our gut and the epithelial surface, beyond which a large amount of our immune system resides. When the mucus layer breaks down, bacteria can come into contact with cells of the gut epithelium, which triggers an inflammatory response that involves the release of zonulin and the opening of tight junctions. This allows the immune system to step in and fix things when they have gone awry. Lectins like PHA and gluten appear to trigger this inflammatory state of leaky gut by interfering with our body's ability to produce a healthy mucus layer as they bind to goblet cells and inhibit their functioning. Gluten is another lectin found in wheat, rye, and barley that is responsible for a huge amount of suffering. It causes celiac disease and lesser-known illnesses such as non-celiac gluten sensitivity. Wheat is full of lectins and also possesses wheat germ agglutinin, WGA. The gliadin molecule is clearly quite toxic to the intestinal cells. This toxicity appears to occur in all humans, not just those with celiac disease, and Dr. Fasano and many other gluten researchers now believe that gluten invariably damages the intestinal epithelium and causes leaky gut. It seems reasonable to hypothesize the following. Lectins in the gut could cause damage there, travel to the brain through the vagus nerve in a retrograde fashion, and trigger injury of dopamine-secreting neurons in the regions of the basal ganglia that control movement and other complex tasks. I strongly believe that inflammation is at the root of most chronic disease, but is manifested differently in each person based on their own unique genetic susceptibilities. Thus, eating lectins and other plant toxins might contribute to Parkinson's disease in some people, heart disease in others, and skin issues or joint pain for others still. The critical error of judgment that Western medicine is making is in imagining that there are thousands of different chronic illnesses, when in fact there's really only one big one: inflammation. Insulin is a hormone released from the pancreas primarily in response to the ingestion of carbohydrates or protein. Its actions in the human body are complex, but in general, insulin is an anti-catabolic hormone that signals our muscles and liver to take up and store glucose. It also signals our fat cells to store fat rather than burn it. Those eating a carnivore diet also note significantly improved satiety and often find benefits beyond those experienced on ketogenic diets, which usually include many foods that are quite high in lectins, such as nuts, seeds, and nightshade vegetables. Lectins have also been found to negatively affect signaling of a satiety hormone known as leptin. In a simplified model, leptin is a hormone that is released when we eat to tell our brain that we've had enough and are full. Plant lectins look like the perfect storm for creating obesity. Not only do they appear to damage the gut, potentially leading to systemic inflammation, they also negatively affect the signaling of hormones such as insulin and leptin, which are centrally involved in regulation of proper body composition and hunger. The foods that appear to be highest in lectins that can trigger our immune system are seeds, grains, nuts, legumes, nightshade vegetables, the skin and seeds of squashes, and traditional dairy products. Since dairy is an animal product, I haven't talked about it yet, but it also contains many proteins, including lectins, which can cause inflammation and immune issues in humans. Lectins are plant weapons. They are designed to discourage predators, and the objective of plants is clear in this case. It is true that some lectins can be denatured with cooking, especially at high pressures and temperatures, but lectins are pervasive in the plant kingdom and occur in many foods that are commonly eaten raw or lightly cooked. It is also true that lectins occur in animal foods as well as in the human body. Reinforcing the concept of disparate operating systems, the majority of research on lectins suggests that plant lectins have the potential to damage our gut and trigger our immune system much more so than those from animal meat and organs. Myth number 1. Plants are superfoods. We debunked many of the myths surrounding supposedly magical chemicals like antioxidants from the isothiocyanate or polyphenol families and showed that these plant toxins can damage DNA, inhibit digestion, disrupt hormonal balance, and trigger the immune system's inflammatory response. We also touched on compounds like lectins and oxalates. The former are common in nightshade plants like goji berries, and the latter are abundant in spinach and many other plant foods often labeled as super. If we harbor any notions that it's the fiber in fruits and vegetables that makes them special, I'll do my best to thoroughly debunk that notion in the next chapter as well. A food must be rich in the micronutrients that humans need to thrive. In addition, these micronutrients must be in the most usable form for human biochemistry, and they must be bioavailable. Cobalamin, or vitamin B12, does not occur in any plant foods, but this is just the tip of the iceberg when it comes to unique nutrients in animal foods. Creatine, carnitine, choline, taurine, and carnosine are also found in appreciable quantities only in small animal foods and are absolutely necessary for optimal health. We can produce a small amount of creatine endogenously on the order of 1 gram per day, but it's not enough for optimal performance of our brains, muscles, or DNA protection and repair. Creatine supplementation had a significant positive effect on both working memory, backward digit span, and intelligence, both tasks that require speed of processing. These findings underline a dynamic and significant role of brain energy capacity in influencing brain performance. Another study of 128 adult females who were supplemented with 20 grams of creatine for 5 days found similar improvements in cognitive function in the subjects who were vegetarians. This suggests that those eating meat already had adequate creatine stores and that a vegetarian diet can result in mental performance deficits due to the lack of this nutrient. Inadequate stores of creatine in the muscle also lead to decreased strength and explosive power. What's important to know is that choline is crucial for kicking butt. A small amount of it can be made in our bodies, but just like creatine, this amount isn't adequate for optimal health. Deficiencies of choline are associated with non-alcoholic fatty liver disease, neurodegenerative disease, and heart disease. The need for this nutrient is also particularly high in pregnant and nursing mothers, and deficiencies have been associated with neural tube defects, poor fetal brain development, premature birth, and preeclampsia. The next C's in this illustrious group are carnitine and carnosine. The Latin root of their names quickly alludes to the fact that these nutrients are almost entirely found in animal meat and organs. Like choline and creatine, our bodies produce some carnitine, but it isn't enough for optimal health. This nutrient helps shuttle fatty acids across the mitochondrial membrane to the interior of this organelle, where they can be used as fuel in the biochemical process of beta oxidation. Carnitine helps us to use our fat stores for energy. This is a very good thing because we have lots of fat to burn, but only a limited supply of available carbohydrates stored in the form of glycogen in our muscles and liver. Using fat for fuel also results in the formation of ketones, which benefit our body in many ways, including turning on the genes associated with longevity, sirtuins and FOXO3, and improving mitochondrial function. Not only were the levels lower in depressed patients, the degree of this deficiency correlated directly with the severity of illness. That is, the levels of carnitine were lowest in those with the worst cases of depression. Mechanistically, lower levels of carnitine are hypothesized to change the way genes in the brain are turned on and off, negatively affecting neurotransmitter levels and causing inflammation. Carnosine is a molecule that acts as an endogenous antioxidant, much like our old friend glutathione. We can make a small amount, but the story remains the same. Multiple studies have found that if we don't get enough through diet, our levels will be suboptimal, and because it's only found in animal foods, vegetarians have repeatedly been shown to possess lower levels. Carnosine appears to be far more than just another antioxidant, however. This little gem of a molecule has also been shown to reduce the formation of advanced glycation end products, AGEs, that can be formed in our body when sugar molecules are added to proteins or lipids. Elevated levels of AGEs have been associated with diabetes, heart disease, and dementia. Vegetarians demonstrate higher levels of AGEs, which may be due to a combination of increased consumption of sugars like fructose and lower levels of protective molecules like carnosine. Taurine is yet another valuable nutrient only found in animal foods that also functions to reduce AGEs. Its functions in the human body aren't fully understood. The pattern here is clear. Animal foods uniquely provide numerous nutrients that we need to kick a lot of butt. Those who eschew these foods invariably suffer from a lower quality of life and unnecessary negative health consequences. The studies noted above encompass more than 130,000 participants and consistently show a correlation between plant-based diets and mental health issues like depression and anxiety. This mountain of evidence suggests that nutritional deficiencies associated with vegetarian diets could be causing issues in the brain. If we truly want to achieve optimal health of our body and mind, high-quality animal foods simply must be a significant portion of our diet. Psychiatric illnesses are no different from chronic illnesses found elsewhere in the body. Both can be related to inflammation and activation of the immune system. We've talked about creatine, choline, carnitine, carnosine, and taurine, all of which demonstrate profound benefits in the human body when present in adequate amounts, and none of which are present in any appreciable quantity in plant foods. In contrast to these magical animal nutrients, minerals like zinc, iron, magnesium, and selenium are found in plant foods, but because of a molecule called phytic acid and that shady character oxalate, our ability to absorb them is significantly reduced relative to animal foods. Both oxalate and phytic acid are used in plants to chelate or bite minerals. These molecules wrap themselves around positively charged atoms like magnesium, phosphorus, zinc, selenium, or calcium to allow for storage within plant cells. The problem is that when we consume phytic acid or oxalate from plants, these molecules can also bind to these minerals in our digestive tracts, preventing their absorption. Predictably, studies of plant-based dieters consistently show lower levels of minerals like iron, zinc, and calcium. Concerns about the nutritional inadequacy of plant-based diets are echoed in a recent review published by the Mayo Clinic. We found that some of these nutrients, which can have implications in neurological disorders, anemia, bone strength, and other health concerns, can be deficient in poorly planned vegan diets. Vegans may be at increased risk for deficiencies in vitamin B12, iron, calcium, vitamin D, omega-3 fatty acids, and protein. Vegans have also been found to have very high rates of iodine deficiency, a problem that could inhibit proper formation of thyroid hormones. Some studies have indicated rates of deficiency as high as 80%. Cells without enough B12 or folate can't divide properly, resulting in megaloblastic anemia, a condition in which red blood cells are much larger than normal. Deficiencies of B12 can also lead to neurological disease, which presents as balance issues and progress to frank dementia. It is important to note that homocysteine levels are known to be elevated more frequently when relying on plant-based nutrition, suggesting impaired methylation. Elevated levels of this compound can indicate a deficiency in many B vitamins, including B12, B6, and folate, and have been correlated with reduced brain size in both healthy individuals and those with Alzheimer's dementia. Some B vitamins like riboflavin or vitamin B2 are very hard to obtain in optimal amounts from plants alone. Riboflavin also plays a key role in the methylation process. There does not appear to be a need for supplemental L-methylfolate to obtain normalized homocysteine levels when there's enough riboflavin in the diet, but in terms of real food, we must obtain this from animal sources. If we choose to get the majority of our foods from plants, it is very likely that in addition to being depleted in minerals, we will also be shortchanged in B vitamins and the fat-soluble vitamins A and K2 as well. Vitamin K is another fat-soluble vitamin that exists in unique plant and animal forms. In plants, vitamin K occurs as K1 or phylloquinone. In animals, it's found as multiple menaquinones, which are named based on the length of their side chain and are collectively known as vitamin K. Researches on the biological activities of K1 versus K2 reveal some intriguing differences between the plant and animal forms of this vitamin. Like beta-carotene, there are no known unique biological roles in humans for vitamin K1. Some texts claim that vitamin K1 is used in the formation of clotting factors, but vitamin K2 can serve this role as well. Vitamin K2, in contrast, is known to have many distinctly important roles in our physiology, including management of proper calcium storage, bone density, and arterial health. Researchers comparing the health benefits of vitamin K1 versus K2 have repeatedly demonstrated that those consuming higher levels of the latter have lower levels of heart disease and cardiovascular complications, while showing no similar benefit from vitamin K1 consumption. These studies stand as a strong testament to the fact that consumption of animal foods is often associated with lower rates of cardiovascular disease, and consumption of nutrients uniquely present in these foods is crucial for optimal health of our arteries. Animals, like humans, use K2 preferentially, and the richest sources of this nutrient are animal meats, organs, and eggs. Foods like liver are a particularly rich source. Natto is a dish made from fermented soybeans that has a good amount of vitamin K2, but in light of the numerous downsides to soy, I believe animal foods are much better sources of this vitamin. Plant-derived protein is nowhere near as usable as animal protein on a gram-per-gram basis. If we want to avoid osteoporosis and maintain the muscle mass that is critical for insulin sensitivity throughout our life, high-quality animal protein is a crucial component of our diet. There are examples of heavily muscled bodybuilders who don't consume animal foods, but in order to maintain this physique, they must ingest tons of highly processed plant protein powders with all of their heavy metal contaminants, lectins, oxalates, and other anti-nutrients. These vegan proteins are less effective at stimulating muscle growth and repair due to their lower leucine content, and they also use much more water to grow ingredients than an equivalent amount of protein from beef. Because we can't synthesize them from scratch, we must obtain them from our diets, and they play vital roles in the membrane of every cell in our body. Omega-3 fatty acids are especially enriched in our brains, with DHA making up 90% of this lipid pool. Deficiencies have been associated with depression, memory and attention deficits, fatigue, and infertility. The problem with trying to obtain omega-3 fatty acids from plants is that this conversion of ALA to EPA and DHA is exceedingly poor. In one study that involved the feeding of 30 grams of ALA-rich flax seeds, or 6 grams of pure ALA, to healthy subjects, there was zero increase in EPA and DHA levels. The best way to ensure optimal levels of omega-3 fatty acids is to obtain preformed EPA, DPA, and DHA from animal foods. Vegan diets do appear to help some people, at least in the short term. One key feature that both the vegan diet and the carnivore diet share is the elimination of certain foods, and it is this elimination which can be incredibly powerful. The mistake in judgment that is often made with regard to plant-based diets, however, is that it is the elimination of animal meat that produces the positive changes. Thousands of stories of those finding vastly improved health on strict carnivore diets or paleo and ketogenic diets that include meat argue strongly against this supposition. In an overzealous attempt to demonize animal foods, many overlook the fact that a vegan diet often excludes mainstream dairy and processed foods. We can't go blaming meat for the damage that the bread, sugar, and junk food have done. At our core, we are humans who became what we are today because we have been eating animals for 3 million years. Access to large amounts of animal foods allowed our brains to grow and our species to thrive over the last 2 million years. From an evolutionary perspective, it makes absolutely zero sense that animal foods would suddenly become bad for us, or bad for some people but not for others. Plant-based diets work in the short term for select people because they have eliminated some foods that were triggering their immune system. In the long term, however, plant-based diets fail due to the lack of bioavailable nutrients or because of the activation of the immune system by many of the plant toxins. Primates must spend nearly all of their waking hours chewing plant matter and must ingest kilogram quantities of it on a daily basis. A caloric deficit may sound like a good thing for someone who is overweight, but for those who are looking to maintain or gain lean muscle mass, plant-based diets are a nightmare, as I found out firsthand. Long-term calorie-limited diets also invariably result in disruption of hormonal balance, and due to the poor bioavailability of protein in plant foods, many of those who use these diets lose valuable lean muscle mass in the process as well. Many of those who try vegan diets notice improvements in the first few months to years, likely due to the elimination of processed foods and sugars, but down the road they are afflicted with significant health issues, malnutrition, and hormonal disturbances. Many examples of elite athletes going vegan only to see their performance decline markedly, often leading them to early retirement. At the highest levels of competition, plant-based diets simply do not allow for optimal performance or durability in athletes. Though there appears to be some variation between humans in terms of how much of any micronutrient we need to function well, these differences are quite small, and most of us need about the same amounts of vitamins, minerals, and fatty acids to allow our beautiful internal workings to flow smoothly. Plants simply don't match up to animals when it comes to providing the nutrients our inner machine needs to run optimally, and there is zero evidence that certain people are better adapted to receiving the nutrients they need from plants. I believe that a nose-to-tail carnivore diet is our fundamental ancestral diet, and that the vast majority of humans on the planet will thrive eating this way. I also believe that some people will be able to tolerate more plant foods than others, and that different plant foods will be triggering for different people. For those with autoimmune illness or another chronic disease, a full carnivore diet might be the best option, but for those who don't seem to be triggered by some less toxic plant foods, these might be included in the diet from time to time. Myth number 2. Fiber is necessary for a healthy gut. Though it seems to have nearly become canon within the medical world, the notion that humans need fiber from plants to have healthy bowel function is simply false. Examination of the literature regarding fiber and constipation quickly illustrates that plant fiber does not lead to better outcomes in patients with this condition. Eating fiber causes those with constipation to have larger bowel movements because there is more material to excrete, but it does not improve any of the unpleasant symptoms of constipation. In fact, it often worsens them. Not only has the addition of plant fiber been shown to have no benefit in constipation, removal of fiber has been shown to improve constipation. When fiber was completely eliminated, 100% of those with constipation had a complete resolution of all of their symptoms. How anyone can claim that plant fiber benefits constipation is beyond me. Because of this study and many others like it, I would go even further to suggest that plant fiber is worsening constipation in many individuals. There are multiple mechanisms by which fiber might worsen constipation, but in many people with imbalances in the gastrointestinal microbiome, fiber appears to promote overgrowth of the wrong types of bacteria in the small intestine. This is a condition known as small intestinal bacterial overgrowth, SIBO. This disorder is often associated with constipation, gas, bloating, intermittent diarrhea, and painful stools. At its root, SIBO appears to be a problem with gut motility. Normally, peristaltic waves pass through the length of our small bowel and sweep down towards the colon, preventing overgrowth of bacteria in the upper portions of our digestive tract. These waves are known as the migrating motor complex and occur every 45 to 180 minutes between meals. In patients with SIBO, the migrating motor complex appears to be hypoactive, allowing the populations of bacteria in the colon to move up into the small intestine, leading to an imbalance and a loss of diversity there. In SIBO, removing plant fiber from the diet is a powerful first step for treating this condition. By not providing the overgrown bugs in the small bowel with fiber, they may gradually recede back to the colon where they belong, and the dysbiosis observed in this condition appears to improve. I believe that most autoimmune disease begins with damage to the gut and subsequent triggering of the large collection of immune cells there. We know that many things can damage the gut and initiate this process, but I think food, and specifically plants, are the main offenders. I believe plants are triggering most of the autoimmune illness we see today, including SIBO, psychiatric disease, and even skin issues like eczema and psoriasis. Diverticulosis is the pathological process that occurs when the innermost layer of the colon, the submucosa, protrudes through the outer muscular layer, forming small pockets that pouch out from the large bowel. Diverticulosis is quite common in Western societies, occurring in more than half of the population in the United States and Canada by the 6th decade of life. It increases the risk of gastrointestinal bleeding, which can be a life-threatening issue. When controlled studies were later done examining this relationship, fiber did not demonstrate a protective role in the occurrence of diverticulosis. Furthermore, as with constipation, some studies suggested possible harm. Compared to individuals with less than 7 bowel movements per week, individuals with more than 15 bowel movements per week had a 70% greater risk of diverticulosis. Neither physical inactivity nor intake of fat or red meat was associated with diverticulosis. A high-fiber diet and increased frequency of bowel movements are associated with greater rather than lower prevalence of diverticulosis. Some have hypothesized that increased pressure in the colon might cause this illness, but this seems unlikely as diverticuli can be found on the right side of the colon, which is low in pressure. The most compelling theory of diverticulosis suggests that it may actually be inflammatory in nature. As we've seen, diverticulosis is incredibly common and increases in incidence with age. There must be some trigger that many are exposed to, which is leading to the gut inflammation that precedes this condition. My money is on this trigger being plants with the many ways that they can damage our gastrointestinal tracts. Perhaps the other most common misconception about fiber is that it lowers the risk of colon cancer. Sadly, this has been repeatedly proven not to be the case. When thinking about the research that has been done on fiber and cancer, we must be careful not to be fooled once again by epidemiology. What we will quickly see when comparing types of research is that while observational trials may show an association between fiber intake and better outcomes, this is likely due to healthy user bias, and interventional trials paint a very different picture. At a molecular level, plant fibers are chains of sugar molecules, polysaccharides, that can't be broken down by our digestion and don't have any nutritional value to humans directly. They pass through our stomach and end up in our small intestines intact. Then they are either broken down by bacteria or pass through us and out into the stool unchanged. Technically, plant fiber is composed of both soluble and insoluble molecules. These terms refer to the ability of the polysaccharide molecules to be dissolved in water. Soluble fibers, like pectin, beta-glucan, and gums, dissolve in water, while insoluble fibers, like cellulose, hemicellulose, and lignans, do not. Psyllium and Aspergilla are examples of soluble fiber, and wheat bran is a commonly used insoluble fiber. Both soluble and insoluble fiber can be used by the bacteria living in our gastrointestinal tract to produce short-chain fatty acids, which are used as an energy source by colonic epithelial cells. The best-known short-chain fatty acid is butyrate, but many others include acetate, isobutyrate, isovalerate, and propionate are also produced by our gut bacteria. The capacity of dietary fiber to bind polyvalent mineral ions, zinc, calcium, magnesium, selenium, iron, may also impart a negative effect on the bioavailability of some nutrients. In effect, the capacity of different fiber-rich vegetables to bind and hold metal ions on their surfaces and thus modify the balance of these cations can be attributed to some of the substances that make up their dietary fiber. There was no consistent evidence for the benefit of dietary fiber with regard to appetite. I don't think these findings should be surprising to us. Appetite, satiety, and loss of fat are much more complex than simply filling our stomachs with non-nutritive plant fibers. In order to effectively create satiety signals, we need to provide our body with a nutrient-rich diet that does not promote massive swings in blood glucose or an excessive release of hunger-promoting hormones. Fiber has failed to show any benefit for constipation, diverticulosis, colon cancer prevention, diabetes, and weight loss. In some cases, there is even evidence that fiber could be worsening these conditions, and there's certainly evidence that many types of fiber and accompanying phytic acid decrease mineral bioavailability. The last bastion of hope that many who champion fiber cling to is the notion that it's necessary for a healthy gastrointestinal microbiome. There is no scientific evidence that we need plant fiber in order to achieve a robust level of diversity in our microbiome. There are studies suggesting that a Western diet is associated with a lower microbial diversity, generally referred to as alpha diversity, but claims that this is causally related to the low fiber aspect of such a way of eating are premature. Diets high in fructose and glucose have clearly demonstrated an ability to negatively alter the microbiome and are much more likely to be some of the culprits in the decline of alpha diversity seen in those eating a Western diet. Let's also not forget about the data we saw in Chapter 7 on lectins, suggesting that these plant toxins can also decrease alpha diversity by allowing overgrowth of facultative aerobes like E. coli. Interventional trials with increased fiber do not reveal increased alpha diversity, and trials with a zero plant fiber carnivore diet do not show decreased alpha diversity. Low fiber ketogenic diets have also been shown to not decrease diversity scores. In a trial following patients with multiple sclerosis for 6 months, the alpha diversity actually increased in those eating a ketogenic diet during this time. There is good evidence that short-chain fatty acids play an important role in the large bowel by acting as a fuel for colonic epithelial cells. The false conventional notion here is that butyrate is the only short-chain fatty acid utilized by these cells, and that in order to obtain this, we must feed the bacteria in our gut with plant fiber. Many different short-chain fatty acids can be used by the colonic epithelium, as can ketones like beta-hydroxybutyrate from within the circulation when eating low-carbohydrate diets. In addition to butyrate, propionate, isobutyrate, isovalerate, and acetate are all short-chain fatty acids produced by bacterial fermentation of protein. In a study examining changes in gut flora with plant-based versus animal-based diets, researchers noted a shift from predominantly butyrate and acetate on a plant-based diet to isobutyrate and isovalerate on a carnivore diet. Short-chain fatty acids are formed within the lumen of the gastrointestinal tract and can be taken up by the epithelial cells of the colon for energy. Within these cells, they go through a series of chemical reactions, transforming them into beta-hydroxybutyrate, a molecule you may recognize as one of the major ketones produced in our bodies when we are in a state of ketosis. Thus, when we choose to eat a ketogenic diet, our colonic epithelial cells can also use the beta-hydroxybutyrate present in our circulation for fuel, lessening the need for short-chain fatty acids from the lumen of the gut. It appears that animals and humans can ferment collagenous tissues from animal meat into short-chain fatty acids. Collagen is the protein that composes most of the connective tissue in our body, including bones, ligaments, tendons, and cartilage. Myth number 3. Red meat will shorten your life. After considering 800 studies, they came to the conclusion that for every 100 grams of red meat eaten per day, there was a 17% increase in colon cancer risk. They also concluded that for every 50 grams of processed red meat, there was a risk increase of 18%. They went on to classify red meat as probably carcinogenic to humans. Only 14 of the 800 studies were considered in their final conclusions, and every single study was observational epidemiology. Of the 14 epidemiology studies that were included in the International Agency for Research on Cancer report, 8 showed no link between the consumption of meat and development of colon cancer. Yes, you read that correctly. The majority of the studies considered in this report did not show a correlation between consumption of red meat and colon cancer. Of the remaining 6 studies, only 1 showed a statistically significant correlation between meat and cancer. Those who were eating red meat tended to engage in other unhealthy behaviors as well. This is an illustration of the concept we've referred to previously as unhealthy user bias, a confounder that often comes into play in studies of those eating red meat. In cultures or religious groups like the Adventists, where the narrative surrounding red meat is negative, those who choose to disregard these ideas generally tend to also do other rebellious and unhealthy things, like smoking, drinking alcohol, and exercising less. In studies of red meat and health outcomes, these quote-unquote rebellious behaviors can be particularly problematic and often skew results. The results of our study suggest decreased rather than increased oxidative stress and inflammation when lean red meat intake is increased at the expense of dietary carbohydrate-rich foods. Our results do not support the suggestion that higher red meat intake leads to increased risk of heart disease and type 2 diabetes via effects of iron to increase oxidative stress and inflammation. For our review of randomized trials on harms and benefits, 12 unique trials enrolling 54,000 participants, we found low to very low certainty evidence that diets lower in unprocessed red meat may have little or no effect on the risk for major cardiometabolic outcomes and cancer mortality and incidence. There is also evidence of possible health benefits of omnivorous versus vegetarian diets on such outcomes as muscle development and anemia. The panel suggests that adults continue current unprocessed red meat consumption. It's still useful to examine the proposed mechanisms by which red meat is hypothesized to cause cancer, and in the process to fully dispose of this farcical notion. These mechanisms include heme iron, N-nitroso compounds, and the heterocyclic amines that may be formed during the cooking process. Studies with heme iron have only shown a potential to induce precancerous lesions in calcium-deficient mouse and rat models. When these animals are fed adequate amounts of calcium, heme iron does not appear damaging to the gut, as was previously demonstrated in the study of beef, bacon, and chicken in rats. As we'll talk about in Chapter 12, a nose-to-tail carnivore diet includes sources of calcium, so contrived rodent models suggesting harm from heme iron are irrelevant. It has also been suggested that heterocyclic amines, HCA, and polycyclic aromatic hydrocarbons, PAH, are mechanisms by which red meat may induce cancer growth in the gut. These can be formed when meat is cooked on a hot surface or exposed to smoke from a fire or grill. I do think it is important to be aware of these compounds and to limit them from our diets as much as possible. Much like our old enemies, the isothiocyanates, these compounds activate the NRF2 system in the liver. It must be detoxified, but our body appears to have plenty of mechanisms to deal with them in moderate quantities. The key word here is moderate. Epidemiology studies have suggested an increased risk of issues with these products of cooking only when they are consumed in very high quantities. Think charred, burned, and massively overcooked meats. Yuck! And who cooks their meat that way anyway? By choosing slow, low-temperature cooking methods, we can easily avoid significant amounts of these compounds in our diets. I'm going to break some hearts here, but I'm not a big fan of grilling and smoking meats for this reason. These are probably fine in moderation, but I think the majority of our food should be prepared with more gentle cooking methods. We don't make new 5-GC due to a mutation in the enzyme that synthesizes it, known as CMAH, but humans do produce new 5-AC, which differs by just one oxygen atom. The loss of function in this enzyme appears to have occurred 2 to 3 million years ago, and is hypothesized to have been a protective adaptation to infectious insults, such as pathogens like bacteria, viruses, and parasites. Such pathogens use the sialic acids on the surface of our cells to attach and gain entry. Some contend that because humans do not possess Neo5G, while ruminants like cows, deer, and lamb do, eating these animals could trigger an immune reaction. There is no evidence in humans to support this idea, however. Humans are not the only species to lack this sialic acid molecule. The mustelids, ferrets, badgers, martens, weasels, etc., are a family of carnivorous animals that also lack Neo5GC, but commonly consume other animals that do possess this molecule. This behavior mirrors the consumption of Neo5GC in red meat by humans. Since we don't see wild ferrets becoming extinct from rampant cancers, and since humans have done nothing to thrive for the last 3 million years eating Neo5GC containing animals, I think it's pretty safe to say that the notion of this molecule being connected with cancer is no less than a fairy tale and not something to worry about. The cells in our body receive inputs from the external environment communicating how they should behave. Sometimes they receive signals that tell them to repair or to perform organized cell death, apoptosis. At other times, like when nutrients are abundant or after exercise, our cells get the signal to proliferate and grow. These two opposing processes of cell breakdown and cell growth are respectively known as catabolism and anabolism, and both serve vital roles throughout our lives as our bodies oscillate between periods of building and recycling of cellular compounds throughout the day. When we eat or exercise, we send anabolic signals to our cells to build and grow. During periods of fasting between meals, our cells receive signals that it's a good time to do some catabolic house cleaning, also known as autophagy. Simply speaking, mTOR is part of a signaling pathway that tells cells to grow and divide. Throughout our lifespan, mTOR is active during periods of rapid growth, like childhood and puberty. During adulthood, it also serves an essential function in maintenance of muscle mass and is turned on in response to resistance exercise. mTOR participates in the anabolic signaling process within our cells in response to 4 distinct signals. These include insulin-like growth factor 1, IGF-1, insulin, protein, mostly mediated through the amino acid leucine, and exercise. Resistance exercise, sleep, carbohydrates, and leucine can all increase mTOR signaling and lead to cellular growth. On the flip side of anabolic mTOR signaling lies the AMP kinase AMPK pathway. This pathway generally serves a catabolic role in our cells and is triggered by the absence of nutrients or growth signals. In a simplified view of our cellular workings, we might think of a seesawing balance between mTOR and AMPK activation. Some cancers harbor mutations in mTOR-associated pathways, leading to excessive cellular growth and proliferation. It has also been noted that in people with Laron syndrome, who possess a defect in growth hormone signaling, there is a low incidence of cancers. Some scientists and physicians have suggested that minimizing mTOR signaling can help us deter cancer. The problems with this interpretation are immediately apparent, however, and it's quite evident that we need mTOR to be strong at healthy humans and to maintain muscle mass as we age. Nevertheless, those fearful of mTOR advocate for dietary limitation of things that might increase IGF-1 or activate mTOR directly, like the amino acid leucine or animal protein in general. Low-protein diets have been repeatedly associated with muscle wasting, a process known as sarcopenia, and are a known major risk factor for mortality. Another often ignored part of the mTOR equation is that this anabolic pathway can be stimulated by both carbohydrates through insulin and protein. Studies comparing anabolic potential show that activation of mTOR by insulin is much more robust and lasts 3 to 4 times as long as its activation by leucine. To suggest that we can lower cancer risk or live longer by limiting animal protein suggests an incomplete understanding of mTOR signaling and ignores millions of years of evolutionary wisdom. Throughout our evolution, we've always had periods of abundance and periods of scarcity. During the times of plenty, our body receives signals to grow from the mTOR pathway, and during times of scarcity, the cellular house cleaning pathways directed by AMPK take over. There have been multiple studies in those consuming high-protein diets, and invariably, these have failed to find a detrimental effect on kidney function. The majority of kidney stones are formed from calcium oxalate, and increased intake of oxalate-containing plant foods appears to be the major risk factor here. Within the medical literature, there's just no clear evidence that higher protein diets are linked to kidney stones. Another common criticism leveled at high-protein diets is that they may lead to lower bone density or osteoporosis due to higher acid load. While it is true that protein in the diet represents an acidic input, this can be balanced by obtaining enough alkalizing minerals such as calcium, magnesium, and potassium. In Chapter 12, we will talk in detail about sources of these minerals on a nose-to-tail carnivore diet that will balance protein consumption from a pH perspective, such as bone broth, bone meal, and bone marrow. In older people with osteoporosis, higher protein intake, equal to or greater than 0.8 grams per kilogram of body weight per day, that is above the current recommended daily allowance, is associated with higher bone mineral density, a slower rate of bone loss, and reduced risk of hip fracture, provided that dietary calcium intakes are adequate. Intervention with dietary protein supplements attenuate age-related bone mineral density decrease and reduced bone turnover marker levels, together with an increase in IGF-1 and a decrease in PTH. There is no evidence that diet-derived acid load is deleterious for bone health. Thus, insufficient dietary protein intakes may be a more severe problem than protein excess in the elderly. If we want strong bones and muscles, a good amount of high-quality animal protein balanced with alkalizing minerals in our diet is the answer. The strongest associations with gout appear to be things that cause insulin resistance, which negatively affects uric acid excretion and likely contribute to disease progression in other ways as well. We must not fall into the trap of blaming meat for what some other damaging food eaten with it has done. If you or someone you know has gout, the best thing that can be done is to eliminate fructose, alcohol, and processed carbohydrates from the diet while enjoying a steak. The concept of blue zones was first suggested by Dan Buettner in a 2005 National Geographic article based on the work he had done with Michael Poulain and Gianni Pes. They described 5 regions of the world where people were observed to live longer than average and suggested reasons for this by looking at similarities in diet and lifestyle between these disparate peoples. The locations included were Okinawa, Sardinia, Loma Linda, California, the Nicoya region of Costa Rica, and Ikaria in Greece. According to Buettner, common to all of these regions were low rates of smoking, a focus on families, social engagement, constant moderate physical activity, and a plant-heavy diet. As the wealth of a country increases, the amount of meat consumed also increases, and wealth has been associated with greater life expectancy. It's possible that part of this trend is due to this association between affluence and meat consumption, but it's also very clear that populations of countries that eat more meat do not appear to be suffering shorter lives in connection with this. DNA contains our genetic code and is wrapped around histone proteins and coiled into chromosomes. Like the tabs at the end of our shoelaces that prevent the ends from becoming frayed, the telomeres cap the ends of DNA, helping to protect the edges of our genetic material from damage. These amazing structures are composed of a repeating series of nucleotide bases and can gradually shorten with every cell division. Investigators use the length of a cell's telomeres to get a sense of its biological age. What sorts of things are associated with longer telomeres? For both vegetarians and non-vegetarians, it is all the great lifestyle stuff we've previously discussed. Exercise, adequate sleep, moderate sunlight exposure, finding meaning in our lives, and a tight-knit community. When it comes to food, there's only one thing that has been correlated with longer telomeres, and that's not plant foods. It's red meat. Among 9 food types, cereal, fruits, vegetables, dairy, red meat, poultry, fish, sweets, and salty snacks, and 8 beverages, juices, coffee, tea, mineral water, alcoholic and sweetened carbonated beverages, only intake of red meat was related to telomere length. Individuals with increased consumption of red meat had higher telomere length, and the strongest significant differences were observed between consumer groups never and 1 to 2 daily. It seems that the central nature of animal foods in the diets of Nicoyans, Sardinians, Okinawans, and Icarians was ignored in the conceptualization of the blue zones. If you have any doubt about this fact, I urge you to visit these places and experience their rich culture, vitality, and zeal for animal foods firsthand. How can it be that the plant-focused diet is resulting in longevity in the Loma Linda community when similar longevity benefits are also seen in steak-loving Mormons in the same state? It's much more likely that the avoidance of destructive behaviors is responsible for the longevity in Loma Linda rather than the increased consumption of plants and decreased consumption of animal foods. Lacto-ovo-vegetarians had lower sperm concentration. Total motility was lower in the lacto-ovo and vegan groups versus non-vegetarians. Vegans had lowest hyperactive motility. The study showed that the vegetables-based food intake decreased sperm quality. Epigenetics are much more important than genetics when it comes to overall quality of life and maintenance of health. Ultimately, the foods we eat and the radical or not so radical way in which we live our life will be the best predictors of how much butt we kick. If there's any magical formula to why people live longer lives, the most consistent commonalities appear to be avoidance of damaging habits like smoking and alcohol, participation in meaningful communities, and favorable genetics. Contrary to the narrative Buettner pitches about the blue zones, in many of these regions as well as other parts of the world where life expectancies are long, meat is quite an important part of the culture and is consumed frequently. Vitamin C, also known as ascorbic acid, is a compound we appear to have stopped synthesizing about 60 million years ago when the primate lineages known as Strepsirrhini and Haplorrhini split, with the latter no longer producing vitamin C. A loss in the ability to break down uric acid also occurred at approximately the same time as these changes in vitamin C synthesis, and some have hypothesized that this molecule may have taken over some of the antioxidant roles of vitamin C as well. I'm not debating that vitamin C serves important roles in our physiology. It's known to be involved in at least 8 enzymatic reactions, including the endogenous formation of carnitine and collagen. Without enough vitamin C, we can't make collagen properly and scurvy develops, with slow wound healing, bleeding gums, skin changes, brittle hair, and loose teeth. Historical accounts, beginning with James Lynn's 1747 report that scurvy in British sailors could be cured with limes, have led us to believe that we must include plants in our diet to obtain enough vitamin C. Fresh meat and animal organs also cure scurvy, a historical fact that has been known for hundreds of years but recently seems forgotten. Contrary to popular belief, animal foods contain vitamin C. Muscle meat has been shown to have approximately 15 milligrams of vitamin C per pound. Organs such as kidney, liver, thymus, and brain are even better sources of this vitamin, possessing 30 to 40 milligrams per 100 gram serving. Similar to the case of vitamin K2, the issue here is that the USDA hasn't formally measured this nutrient in meat and organs, and as a result it is often reported as being zero, but it certainly is not. Vitamin C also appears to be more heat stable in animal foods than plant foods, so cooking meat and organs will likely not result in as much loss of this nutrient. By completely withholding vitamin C in the diet, the first symptoms of scurvy developed within 2 months in these detainees. When doses of either 10, 30, or 70 milligrams of vitamin C were provided, all groups recovered in days with no notable clinical differences between them. Though vitamin C does have roles in our body beyond enabling proper formation of collagen, it's unclear that doses beyond those that are known to correct scurvy are of any further benefit. Interventional studies with supplemental doses of vitamin C have repeatedly failed to show benefit in the endpoints of total mortality, cardiovascular disease, blood pressure, or incidence of the common cold. A 12-week intervention with increased fruits and vegetables was not associated with effects on antioxidant status or lymphocyte DNA damage. There is also evidence that too much vitamin C can be harmful in humans, with reports of an increased incidence of oxalate kidney stones, nausea, bloating, acid reflux, B deficiency, and even increased oxidative stress. When given in large doses, vitamin C appears to turn into a pro-oxidant in our bodies, and even moderate doses of vitamin C found in common supplements, 500 milligrams to 1000 milligrams, have been linked with increased rates of kidney stones. Contrary to the prevailing thinking, I believe that throughout this time our main sources of vitamin C would have been from animal foods, since plant sources would have been scarce at times, depending on the time of year and the latitude. Red meat or animal foods causing cancer or shortening our lives is evolutionarily inconsistent. These are the foods that made us human and the foundational foods that our diets have been based on for the last 2 million years. Eating these foods has allowed us to grow much larger brains and to become a smarter, stronger, and more resourceful species. Myth number 4. Red meat causes the heart to explode. Few things conjure more fear in the hearts and minds of the general population than the big bad cholesterol monster and the associated trepidation that red meat will cause your arteries to become filled with plaque. After all, we've been told by cardiac surgeons that when they scoop plaque out of the arteries in our heart or neck, it looks just like animal fat, eggs, or butter. The word cholesterol is often used colloquially to refer to all of the lipoproteins in our blood, but technically cholesterol is a steroid backbone type of molecule that is used to make all sorts of vital compounds in human physiology. Our body makes around 1200 milligrams of cholesterol every day for many important purposes, including proper formation of all of our cell membranes. The cholesterol molecule is also used as a precursor for all of the steroid hormones in our body. These include estrogen, testosterone, cortisol, progesterone, and aldosterone. The bile acids used to help digest fats are made from cholesterol as well. Without them, we would quickly become malnourished and deficient in fat-soluble vitamins like A, K2, and E. When our skin is exposed to sunlight, cholesterol is also a precursor for the formation of vitamin D. Because cholesterol isn't soluble in the water-based aqueous environment of our bloodstream, our body uses a lipoprotein transportation system to shuttle it between the liver and the various tissues where it is needed. The fat we eat is absorbed from our intestines and packaged as triglycerides with dietary cholesterol into a type of lipoprotein known as chylomicrons. VLDL particles are sent out into the peripheral circulation, delivering cholesterol and triglycerides to peripheral cells. As triglycerides are extracted from VLDL, these lipoproteins contain a higher percentage of cholesterol and increase in density, becoming intermediate and then low-density lipoproteins, IDL and LDL. LDL particles therefore contain both triglycerides and cholesterol as they circulate throughout our bloodstream. The main role of HDL is to take extra cholesterol back to the liver in a process known as reverse cholesterol transport. But as we'll see later, HDL and other lipoproteins also serve vital roles in the immune response. We might think of this whole process of the movement of cholesterol and triglycerides throughout the body as a bus system with various routes and different buses carrying passengers throughout these. Buses carrying dietary fat and cholesterol, called chylomicrons, depart from the intestines to the liver, which is basically the central bus station. Here, passengers get off these chylomicron bus and get on the departing VLDL buses, where they are joined by new passengers from the liver. The VLDL bus makes stops throughout the body and eventually has many less passengers. Along the way, it becomes the LDL bus, which continues on its routes before eventually returning to the liver. The HDL bus starts empty in the liver or intestines and picks up passengers from cells of the body or other buses, taking them back to the central liver bus station. This is an oversimplification, of course, and the lipoproteins in our bodies do much more than simply shuttle fat and cholesterol around our body, but at a basic level, this model describes how the lipoprotein particles move and interact. Most of the cells of our bodies can make a bit of cholesterol from scratch, but they also rely heavily on the delivered supply of this molecule to build membranes and hormones. In the ovaries and testicles, for instance, cholesterol delivered by LDL is necessary to make the lovely sex hormones estrogen and testosterone, which are crucial for libido and reproductive function. In addition to its vital role transporting building blocks and nutrients, LDL also serves important roles within the immune system. Mice with experimentally increased levels of LDL are 8 times more resistant to endotoxin. As a result, they have significantly delayed mortality when directly injected with gram-negative bacteria. Conversely, rats bred to have decreased levels of LDL had much higher mortality rates and levels of inflammation when injected with endotoxin, deficits that could be remedied by providing them with an exogenous supplemental LDL. There are many studies that show elevated levels of LDL are not a risk factor for increased all-cause mortality or cardiovascular mortality in the elderly. Furthermore, there are many studies suggesting that higher levels of LDL are protective as we age, which is most likely connected with its role in immune function. Mortality from cancer and infection was significantly lower among the participants in the highest total cholesterol category than in the other categories, which largely explained the lower all-cause mortality in this category. In people older than 85 years, high total cholesterol concentrations are associated with longevity owing to lower mortality from cancer and infection. It seems pretty safe to say this association is real and that LDL appears to be quite protective as we age. LDL itself is not harmful, but in certain situations it can be involved in the process of responding to injury and inflammation, making it look like it's a bad actor when it's merely present at the scene of the crime. LDL is present in atherosclerotic plaques, often having been ingested by immune cells known as macrophages within the arterial wall. Considered together with epidemiology studies showing a correlation between LDL and cardiovascular disease, this finding has led many to believe that elevated levels of LDL are to blame for this initiation and progression of atherosclerosis. If we examine plaque formation in detail, however, we'll see that it's far from proven that the LDL molecule is intrinsically damaging to our arteries or is enough to initiate plaque formation within the arterial wall on its own. The walls of both veins and arteries are composed of multiple layers. The single-cell layer closest to circulating blood is known as the endothelium, below which lie the cells of the intima, and then a layer of smooth muscle cells. The currently accepted paradigm of plaque formation is known as the response to retention hypothesis. According to this theory, LDL in our bloodstream passes through the endothelium of the artery wall and can become retained or stuck in the intimal cell layer below. When LDL gets stuck in the arterial wall, it may become oxidized, altering the structure of the ApoB100 molecules in its membrane and triggering an immune response in which macrophages within the intima ingest LDL, become lipid-laden foam cells. This is thought to be the beginning of an atherosclerotic plaque. Therefore, according to the response to retention theory, the greater the amount of LDL in circulation, the greater the risk that it will be retained in the subendothelial space and lead to plaque formation. This key portion of the theory has not been proven. There is exactly zero research that conclusively shows that the amount of LDL retained is directly linked to the amount of LDL in the circulation, and there are many studies that suggest exactly the opposite. In the elderly, higher levels of LDL appear to be protective and are associated with greater longevity. Epidemiology studies of women, Canadian and Russian men, Maoris, and Asians do not show associations between total cholesterol or LDL levels and incidence of heart disease or all-cause mortality. If the process of atherosclerosis were really as simple as more LDL in the circulation leading to more plaque formation, this correlation would be present across all age groups, sexes, and cultures. Vegetarians are also far from immune to atherosclerosis. They too display rates equivalent to the general population despite lower average levels of LDL. Furthermore, in statin trials, there is not a true dose-response relationship between levels of LDL and the degree of plaque formation. The progression of atherosclerosis depends on more than simply the amount of LDL in the bloodstream. Another very interesting thing about HDL is that its level correlates directly with insulin sensitivity. In those with insulin resistance, HDL levels fall and triglycerides levels rise, a change in blood parameters known as metabolic dyslipidemia. Thus, those with the lowest levels of HDL are much more likely to have insulin resistance than those with high levels of this lipoprotein. Differences in HDL make a huge difference in cardiovascular disease risk. With low HDL, less than 45 mg per deciliter, there is a clear correlation between total cholesterol and heart disease, but in those with higher levels of HDL, this correlation vanishes almost entirely. If we are insulin resistant, higher levels of LDL may very well contribute to plaque formation and progression, but if we are insulin sensitive, higher levels of LDL are not associated with increased atherosclerosis and are likely protective. Levels of LDL in the blood do not consistently correlate with the progression of coronary artery disease in the absence of insulin resistance. Every second of every day, lipoproteins like LDL are moving in and out of the walls of both veins and arteries, delivering nutrients to the cells there for energy and the construction of membranes. Clearly, there must be another part of this equation that leads to retention of some of these LDL particles within the arterial wall. Unless LDL and the intimal space are sticky, no matter how many LDL particles we have floating around our bodies, the LDL balls only get stuck to the intimal walls when both are coated in a molecular Velcro that makes them sticky. LDL is not enough to initiate atherosclerosis on its own. It has to get stuck in the arterial wall to participate in this process. During the states of insulin resistance and inflammation, both the LDL particle and the intimal space get coated in molecular Velcro and become more sticky. There is evidence that a whopping 88% of the American population has some degree of metabolic dysfunction. One of the biggest mistakes Western medicine makes is to extrapolate these pathologies to the 12% of us who are not insulin resistant and not inflamed, warning us that certain cardiovascular disease will swiftly follow with an elevated LDL. Those of us with good insulin sensitivity are essentially a different breed, and there are many striking stories of plaque regression among insulin-sensitive individuals with elevated LDL eating carnivore or ketogenic diets. In the absence of insulin resistance and inflammation, higher levels of LDL are probably protective because of their roles with the immune response. There is good evidence to believe that LDL is not causing atherosclerosis, but may be involved in plaques as part of a repair process in response to injury to the vessel wall. The endothelium of our veins is exposed to exactly the same amount of LDL as the endothelium of our arteries, and yet only the latter display atherosclerotic lesions. A clue as to why this occurs is found in the common location of plaque buildup at arterial branch points, where turbulent blood flow can damage the lining of our arteries. It appears that atherosclerotic plaques occur in locations where the endothelium has become damaged. This damage can occur as a result of turbulent blood flow, inflammation, insulin resistance, or other causes. When everything is copacetic, these lipoproteins move into the intimal cell layer and then move back out into the circulation, continuing their journey and eventually ending up back at the liver. But when the arterial wall is damaged by states of insulin resistance, inflammation, oxidative stress, hyperglycemia, or arterial turbulence, it displays increased levels of proteoglycan molecules and becomes stickier. This traps the circulating lipoproteins that may also be coated in molecular Velcro. Once stuck in the deepest layers of the intima, LDL is more likely to become oxidized and engulfed by the macrophages there, leading to the beginnings of atherosclerosis. Within this framework, the accumulation of lipoproteins within the arterial wall might be viewed as a molecular repair mechanism. It's not the LDL that initiates plaque formation, it's injury to the arterial wall. The notion that LDL deposition may in fact be part of an arterial repair mechanism is strengthened by the fact that all placental mammals, including herbivores, omnivores, and carnivores, are known to demonstrate LDL deposition in the arterial wall. Although there is no formal characterization of insulin resistance, it's generally defined as a diminished response to the actions of insulin by all of the tissues of the body. Insulin is a peptide hormone secreted by the beta cells of the pancreas in response to protein or carbohydrate intake, but more so with the latter. It signals the peripheral tissues to take up glucose from the bloodstream and to proliferate. When cells of the body become resistant to insulin, more is needed to achieve the previous effect, and blood levels begin to rise. Insulin resistance is essentially synonymous with prediabetes. The main issue is usually excessive food consumption, especially excess carbohydrates combined with fat. Intriguingly, overfeeding with each of these macronutrients individually does not appear to lead to insulin resistance or weight gain, but when they are consumed together in excess, it appears to cause negative metabolic consequences in our body. In situations of insulin resistance, one of the most effective therapeutic interventions is to significantly limit either carbohydrates or fat from our diet until we have lost excess weight and regained proper insulin sensitivity. Since this is a book about the carnivore diet, you might be able to guess that I favor a high-fat, low-carbohydrate approach to insulin resistance related to obesity, but a high-carbohydrate, low-fat approach may also work for some people. In a sinister recapitulation of the unique evolutionary combination of fat and carbohydrates in breast milk, candy bars, ice cream, and many other junk foods similarly function to short-circuit normal mechanisms of satiety and induce rapid weight gain and insulin resistance when eaten as part of a caloric surplus. Our ancestors would have sought out the fattiest animals, but it's unlikely that they always had a lot of fat in their diet unless their hunts were successful. Carbohydrates are similarly scarce within the natural world. Depending on latitude and that time of year, a small amount of fruit may have been available occasionally, but this was likely rare. It's also interesting to note that fructose, the 5-carbon sugar prevalent in fruit, has been shown to promote both insulin and leptin, a satiety hormone, resistance when consumed in excess. Though overfeeding is the first thing we usually think of with regard to obesity and insulin resistance, there are other things that can also cause this metabolic derangement, including infections, inflammation, and lifestyle factors such as stress and lack of sleep. I have included tests that give us a sense of insulin sensitivity. These include fasting glucose, fasting insulin, C-peptide, fructosamine, and fasting leptin. Though fasting glucose is commonly ordered, the others are rarely looked at, but are some of the most valuable data points we can gather. Triglyceride and HDL levels also give a good indication of insulin sensitivity. I prefer to see triglycerides less than 75 mg per deciliter and view a triglyceride to HDL ratio of less than 1 as a good indicator that someone is likely insulin sensitive. The best way to get a sense of our average blood sugar is to use a continuous glucose monitor, CGM. These monitors are quite valuable as they can also show us how blood glucose levels change postprandially after eating and throughout the day and night. When postprandial glucose levels are elevated, it is a signal that some degree of insulin resistance or metabolic dysregulation is occurring in response to specific foods. Research suggests that glucose excursions from baseline should not be more than 50 mg per deciliter, and that less than 30 mg per deciliter is probably ideal. Furthermore, there should be a return to baseline within 1 to 2 hours. I'm not saying that all carbohydrates are bad. Clearly, at times throughout our evolution, we would have eaten some fruit or tubers if we needed to, but I do not believe these made up a significant proportion of our diets. There are indigenous peoples who eat significant amounts of carbohydrates and maintain good health, but there are also examples of groups like the Pima Indians, in whom a high carbohydrate diet has led to an enormous rate of diabetes, greater than 80%, with profound negative health consequences. Elevated LDL is only a problem in the presence of insulin resistance, and if we don't have sticky LDL molecules in arterial walls, more is probably a good thing. Throughout our evolution, it is certain that our ancestors experienced prolonged periods of calorie deficits or complete caloric deprivation when hunts were not successful and there were few planned fallback foods available. Does it even make an iota of sense that our bodies would have evolved a system in which a molecule that is bad for us, like so many claim LDL to be, would increase significantly during such times? Elevated LDL on its own does not appear to be contributing to the progression of atherosclerosis. Similarly, LDL that rises in the setting of ketogenic physiology is very unlikely to contribute to plaque progression and isn't something to be concerned about. If we chose to begin a ketogenic diet like most versions of the carnivore diet, it's certainly possible that our LDL levels will rise. I'm not saying that statins do not decrease cardiovascular mortality. They certainly have been shown to do this, but it's not happening by lowering LDL. As we've discussed earlier in this chapter, there's no dose-response relationship between LDL lowering with statins and improved outcomes, and many other drugs not in the statin class of medications that also lower LDL have repeatedly failed to show any benefits in terms of cardiovascular outcomes. Statins have other pleiotropic effects, including being anti-inflammatory and immunomodulatory, which likely account for the small but significant improvements in cardiovascular endpoints observed in studies. The fundamental flaw of epidemiology studies looking at trimethylamine and oxide levels and cardiovascular disease is that although they might show correlation, they are blind in knowing what direction the arrow of causality might go. That is, epidemiology studies that show correlation between TMAO and these diseases can't show us if TMAO is causing diabetes and cardiovascular disease or if it's the other way around. Quote, "Our findings support that T2DM and kidney disease increase TMAO levels, and observational evidence for cardiovascular diseases may be due to confounding or reverse causality." Unquote. The notion that saturated fat is bad for us originated in the early 1960s with the work of Ancel Keys, who published the now infamous 7-country study looking at the relationship between diet and heart disease in cohorts of people living in the US, Japan, Yugoslavia, Greece, Italy, the Netherlands, and Finland. This observational study showed a correlation between the amount of saturated fat consumed in the diet with blood cholesterol levels and the incidence of heart disease. This gave rise to the diet-heart hypothesis, which postulated that saturated fat in our diet raises cholesterol levels and leads to heart disease. More people are beginning to wake up to the fact that saturated fat is not the outlaw it's been labeled as. We've been eating animal foods with saturated fat in them for the entirety of our evolution as humans. This has led us to become the extremely intelligent, large-brained, strong, and adaptable people we are today. Science has also recently begun to support the things we already knew to be true, and studies have shown that high-fat ketogenic diets containing plenty of saturated fat reverse diabetes and insulin resistance. They also lead to weight loss and improvements in inflammatory markers, as well as a reduction in hypertension, dementia, polycystic ovarian syndrome, and a host of other conditions. Animal studies show that saturated fat does not induce leaky gut, but polyunsaturated vegetable oils like corn oil do open tight junctions and damage the gut lining. The more animal fat and protein people ate, the less cardiovascular disease they experienced, while those who ate the most carbohydrates had the highest rates of this pathology. Hopefully, this time we can keep agribusiness interests out of this process. It's not saturated fat or animal meat that are causing heart disease, it's processed vegetable oils and processed carbohydrates that are the real malefactors here. What to eat on a nose-to-tail carnivore diet. We saw that stable isotope analyses of fossils strongly suggests that our ancestors were high-level carnivores. They ate mostly large animals and displayed higher levels of delta N15 nitrogen than those of contemporary carnivores like hyenas. We also dug into the data regarding duplications in the salivary amylase gene and discussed why the fact that only Homo sapiens possess this suggests strongly that we have not had many starchy foods in our diet for the majority of our evolution. An examination of the size of the human brain revealed the striking finding that about 2 million years ago our noggins started increasing in size dramatically, right when we began hunting animals and using stone tools and weapons. This trend continued up until about 40,000 years ago. No one knows why our brains have been shrinking since then, but it's intriguing to note that this point in our history correlates with the advent of agriculture and a decrease in our access to B12 and other nutrients vital for brain health found predominantly in animal foods. Vegans often deride the act of killing and eating animals as cruel, but nothing could be further from the truth. Few things in my life have been as moving as hunting an animal in nature. Stalking deer with my bow required me to be in the wilderness for long periods of time, something that changed any human who experiences such a communion with the natural world. When killing an incredibly beautiful deer, I felt an immediate responsibility to honor the life of that animal by being the best human I could be. I was reminded of this every time I ate the meat and organs that I had procured for myself. Sadly, I don't get the opportunity to hunt as much of my food as I'd like. Ideally, all of my food would come from animals that had been respectfully hunted, but for now I obtain some meat and organs from a butcher or grocery store. When I do this, I am struck by how separated I feel from the animals that nourish my body and soul, and am reminded of how different it felt to hunt my own food. By understanding where our food comes from, we realize that in order for something to live, something else must die. This is the way of life. By being a participant within this beautiful cycle of death and life, we all bear the responsibility to live a compassionate and kind life that comes with it. The large number of animals killed in the process of harvesting plants, with large machines that kill small animals like rabbits, mice, and other rodents, far outweighs the loss of life when we consume animals directly. The disruption of ecosystems with monocrop agriculture also disrupts an even greater number of organisms on a long-term basis. Any intentional change we choose to make in our diet that includes a good amount of animal products and eliminates some plants will result in significant improvements to our health. I realize that the way I eat isn't going to be the best fit for everyone reading this book, so in an effort to make the carnivore diet as accessible as it can be, I've created 5 tiers of carnivore eating. The diets that I describe within all of these tiers are amazing in my eyes. My advice is to find what works best for you and to begin your personal journey there. A Tier 1 carnivore diet emphasizes animal food consumption as the majority of the diet, but it also allows some room for the least toxic plant foods. Because animal foods represent the most nutrient-rich sources of bioavailable vitamins and minerals, they form the majority of the diet. Approximately 80 to 90% of the diet will be animal foods and might include beef, bison, lamb, poultry, and fish. It could also include eggs and certain dairy items. In addition to these foods, low toxicity plant foods may be included. I generally think of the least offensive plant foods as non-sweet fruits, including things like winter or summer squash, avocados, cucumbers without skin or seeds, and olives, as well as seasonal berries. At the opposite end of the spectrum are seeds, grains, nuts, and beans. These are all plant seeds, and they are all very heavily defended by plant toxins. Artificial sweeteners of both plant and synthetic origin have no place in a healthy diet and are probably going to sabotage our efforts at weight loss, satiety, and overall improved health. Many spices are also made from the seeds of plants and are best avoided on any type of carnivore diet. These include black and white pepper, coriander, cumin, cardamom, nutmeg, cloves, mustard, and caraway. Cinnamon is made from the bark of a tree and can also cause immune reactions in some people. Spices made from plant leaves are often referred to as herbs and might be more tolerable than seed-based spices, depending on the individual. These include oregano, basil, rosemary, parsley, dill, sage, and mint, all of which might be a part of a carnivore diet for flavor or variety, but I don't believe they have any unique benefit. In terms of flavoring for food, the best place for most people to start is a good sea salt. When transitioning to a ketogenic diet or fasting, it is important to keep an eye on salt intake, and most people feel best when they are getting 6 to 10 grams of salt per day. We discussed oxalates in detail and described ways in which they can contribute to kidney stones, chronic pain, and many other negative effects in the body. You may want to refer back to that chapter for a chart showing some of the highest oxalate-containing foods, but the worst offenders are spinach, turmeric, almonds, potatoes, navy beans, and beets. The nightshade family also resides on the toxic end of the spectrum and is known to be a common immune trigger. This family of vegetable includes tomatoes, eggplants, white potatoes, goji berries, bell peppers, paprika, and chili peppers. As we've seen previously, foods from this group have been shown to open tight junctions in the gastrointestinal tract, creating leaky gut. Give these a pass. Based on their content of isothiocyanates, I'd also place brassica vegetables far to the right on the spectrum of plant toxicity. I've placed most fruit on the bad side of the plant toxicity spectrum. In this graphic, sweet fruit is basically all of the fruit we traditionally think of except berries, which we'll discuss a bit later. This includes things like apples, pears, grapes, mangoes, and so on. At first glance, these types of fruit seem like something our ancestors would have eaten when they were available, and they probably did from time to time. But they did so only for very limited portions of the year and in very specific parts of the world. Think about the forests, deserts, plains, and other wild spaces near where we live. How often do we see fruit growing out in the wild? Rarely. Closer to the equator, fruit is more available, but still occurs seasonally. The vast array of fruit that we see in grocery stores every day of the year is an incredibly inaccurate representation of the availability of these foods in the natural world. There is nowhere on the planet that things like melon, mango, berries, apples, and citrus fruit all grow wild in the same place or year-round. Over the last 100 years, through hybridization practices, most of the fruit we eat has been made much sweeter and larger than its wild cousins. Aside from the lack of beneficial effects, there are also concerns that fructose and glucose in fruit might be bad for us. Excess consumption of both of these sugar molecules have been shown to have all sorts of harmful effects in the human body. Overindulgence in fruit is also linked with changes in pH of the mouth, which can lead to tooth decay. The dental benefits of a low-carbohydrate diet like the carnivore diet are well documented, with studies showing reductions in tooth decay and types of gingivitis-promoting bacteria like P. gingivitis that have been linked to Alzheimer's disease. There are also numerous reports from those eating a carnivore diet of reversal of gingival disease and recession of the gums, further evidence that when constructed correctly, this way of eating provides an adequate amount of vitamin C. Beyond sheer calories, fruit doesn't have much redeeming nutritional value. It's just a plant's way of getting us to do it a favor and move its seeds around to fertile locations. Honey is another fructose-rich food that probably should not make up a significant proportion of any healthy diet. Evidence of tooth decay in avid honey eaters within certain African tribes and Pacific Islanders stands as further testament of the potential dental downsides to overconsumption of sugar-rich food. But Dr. Paul Saladino now regularly consumes honey. In the middle of the spectrum of plant toxicity, we find tubers and berries. Though berries are technically a fruit due to their lower sugar content, I'd consider them to be a bit less likely to cause issues than the sweet fruit. Have you ever eaten a wild carrot? They are about half the size of your pinky finger. Expending energy looking for food like this would have been a very bad use of time if other more nutrient and calorie-rich foods were available. Some proponents of higher carbohydrate paleo diets have argued that modern hunter-gatherers like the Hadza or Kung consume about half of their calories from tubers. But as many anthropologists have pointed out, these people have been marginalized and forced to change their hunting practices, no longer serving as accurate representations of the generations of indigenous peoples that preceded them. These groups are now forced to live on smaller areas of land and are no longer allowed to hunt the elephants and other large game that their ancestors relied upon for thousands of years. Fungi are a separate kingdom from plants, but they do still make many toxins to defend themselves against animal predation. Working with clients in my private practice, few things elicit more heartfelt groans of agony than the suggestion that they should eliminate coffee from their diets. Those that are able to make this lifestyle change, however, report more stable energy levels, better sleep, and brighter overall moods. Though it would take a heck of a lot of coffee to kill you, about 75 cups, caffeine does have a lethal dose in humans. Drinking a few cups of coffee per day may result in an addiction, however, and can exert negative physiologic effects in humans, including raising heart rate, blood pressure, blood glucose, and the subjective experience of acute anxiety. Caffeine addiction has also been linked to chronic depression and anxiety, and withdrawal from this substance can be quite unpleasant. Acrylamide is yet another downside of coffee. It is formed during the roasting of many foods, including nuts, crackers, bread, breakfast cereals, French fries, and potato chips, and is also found in cigarette smoke. So when it comes to beverages on a carnivore diet, what should we drink? Pardon me if this sounds pedestrian, but how about good old water? This is what our ancestors have been drinking for the last 4 million years, with perhaps a bit of blood mixed in there as well. It is worthwhile to consider drinking higher quality water than the fluoride, chlorine, and pharmaceutical enriched liquid that comes straight out of our faucets. We debunked the myth that resveratrol has a valuable role in our diet, and after the previous discussion of mycotoxins, we've further seen that the notion that wine is beneficial for us is largely a misconception. Finally, much like coffee, conventionally grown grapes are heavily sprayed with pesticides, and many wines have added sulfides. No wonder so many of us suffer from raging headaches after drinking the red stuff. Distilled spirits like vodka or whiskey might contain less of the contaminants that can negatively affect some people. They would probably be the cleanest version of alcohol to drink if we choose to indulge in moderation from time to time, but beer and wine are off the team. Eat when you are hungry and consider an eating window most days. I do think the time-restricted eating principle of having a confined eating window with a longer period of fasting is a valuable addition to your lifestyle with a carnivore diet. Very often, people end up eating fewer times throughout the day on this type of diet because they aren't as hungry. After 1.5 years of eating a nose-to-tail carnivore diet, I don't even know how I ever ate 3 or more times a day. Moving the eating window earlier in the day, also known as ETRF, has been shown to result in improvement in many markers. On a Tier 2 carnivore diet, eat meat, drink water is the classic adage. It's a pretty simple formula, and as an elimination diet it can be a very powerful tool. Though there are a few examples of people doing well long term on a diet such as this, not everyone appears to thrive when eating this way. My main concern beyond short-term use is the possibility of nutrient deficiencies. While I do think a Tier 2 carnivore diet can be very helpful for some people, adding a few foods like eggs and occasional seafood will help fill in many of the potential nutrient gaps. Evolutionarily, I also don't believe that we would have eaten only the muscle meat of animals. There are numerous examples from anthropological literature to suggest that indigenous groups consistently prized organ meats. A Tier 3 carnivore diet includes meat, eggs, seafood, and dairy, if tolerated. Eggs alone enrich a carnivore diet by adding good amounts of vitamin A, choline, vitamin K2, DHA, and folate, but there still might be some nutritional holes on a Tier 3 carnivore diet. Seafood is another valuable part of a carnivore diet. Low mercury fish like wild salmon and sardines will be good sources of EPA and DHA, as well as the iodine needed to make thyroid hormones. Shellfish are also incredibly nutrient-rich, with oysters being superheroes when it comes to zinc and omega-3, and mussels are loaded with manganese. When thinking about seafood, it's also important to remember that humans have sadly polluted oceans and lakes, and in general, both freshwater and saltwater seafood may carry a higher toxic burden than freely grazing land animals. Larger fish like tuna, halibut, king mackerel, swordfish, and shark contain very high levels of mercury and other heavy metals and should be avoided completely. Wild salmon will be much lower in this regard and may be eaten once or twice a week if desired. Shellfish can have higher levels of cadmium and other heavy metals as well. Eating mussels, oysters, clams, shrimp, crab, lobster, or scallops once or twice a week is a great addition to a carnivore diet. Most people find that ruminant animals like beef, buffalo, lamb, and venison are more satisfying and enjoyable. From a nutritional perspective, ruminant muscle meat and organs are generally better sources of the vitamins and minerals we need. What an animal eats affects the quality of its meat and organs. Do we really want to be eating animals fed with grains contaminated with pesticides and mold toxins? For those looking to dip a toe into the waters of including organ meats in their diets, liver and heart are a great place to start. By adding these, we'll significantly level up on nutrient adequacy. Heart is similar to muscle meat in nutritional content, but has a bit more CoQ10. It's got a slightly different flavor and is tender if cooked properly. The liver doesn't store toxins, it chemically transforms them. This is how we get rid of the nasty chemicals and compounds hanging around in our bodies. Liver is particularly rich in many minerals and B vitamins that complement those found in muscle meat. B vitamins are found in robust amounts in organ meats like liver and kidney, but levels are much lower in muscle meat. If we don't include organs in our diet, we can become deficient in these nutrients, leading to rising homocysteine levels and possible issues with methylation reactions within detoxification, neurotransmitter synthesis, and hundreds of other biochemical processes in the body. Eating raw egg white is a bad idea. In raw egg whites is a compound called avidin that can bind biotin and cause a deficiency of this nutrient. The RDA for biotin is 30 micrograms per day, which is present in a few ounces of liver or kidney, but we'd have to consume 21 ounces of steak to get this amount. While liver from ruminants is a very nutrient-rich food and much lower in vitamin A than polar bear liver, approximately 180 international units per gram versus 20,000, it's probably best not to consume excessive amounts of it until this vitamin A research is further fleshed out. For most people, 8 to 16 ounces of liver per week will be totally safe in terms of vitamin A, and it will provide robust doses of all the other valuable nutrients present in this organ. Muscle meat is rich in methionine and connective tissue rich in glycine. Though we don't always see it in the grocery store, animals are about half muscle and half connective tissue. We should probably eat a reasonable amount of each in whatever form is easiest on a daily basis if we want to mimic the way we've been eating animals throughout our evolution and ensure optimal performance of our biochemical engines. A Tier 5 carnivore diet builds on the foundation we've already put in place with the previous tiers, including grass-fed muscle meat, connective tissue, liver, seafood, and eggs. It also adds in more organ meats and considers fat to protein ratios based on performance goals. They don't exactly taste like a Snickers bar, but kidneys have as much riboflavin, K2, folate, and vitamin C as liver. Exploration of organ meats beyond kidneys is definitely for the adventurous. Those who choose to consume them have generally found they feel better overall and have more energy when including a larger variety of these foods in their diet. In a given week, I might eat brain, spleen, pancreas, thymus, and quote-unquote oysters, testicles, in addition to my staples of liver and kidney. We can really only obtain about 40% of our calories from protein before it starts to put stress on our biochemistry. We don't use amino acids directly for fuel. We convert them to glucose through gluconeogenesis when the need arises. Protein is best thought of as a building block, while fat and carbohydrates fuel the metabolic engine. Once we've obtained enough protein to grow and repair the structural components of our body, more of this macronutrient doesn't seem to be helpful, and too much might tax our system in the excretion process of excess nitrogen groups from amino acids. The benefits of a ketogenic, fat-based metabolism are many. There is evidence that such diets can reverse insulin resistance and diabetes, lower blood pressure, increase mitochondrial biogenesis, turn on longevity genes, and reduce oxidative stress and DNA damage. There is further evidence that these diets can decrease appetite, lead to profound weight loss, improve mood, and be protective for our brain. From a cardiovascular risk standpoint, a ketogenic diet has also been shown to decrease triglycerides and metabolic dyslipidemia while increasing HDL. On the other hand, low-fat diets generally increase triglycerides, lower HDL, and raise levels of insulin. This will be different from person to person, but once we design a dietary framework that gets us the Goldilocks amounts of protein from meat and organs, the rest of our calories will come from fat. I generally recommend a 1-to-1 ratio of fat to protein by grams for most people, but some will eat even more fat than this. It's completely possible to lose weight eating fat in a 1-to-1 ratio with protein on any of the types of carnivore diets I have described. The secret to this is in how satiating fat is as a macronutrient. Once we start incorporating more good fat sources in our diet, we may be surprised at how full we feel throughout the day. In this situation, it's incredibly freeing to no longer be controlled by our appetite all of the time, as so many are on carbohydrate-heavy diets. Animal fat is a great source of fat-soluble vitamins like E and K2. Grass-fed animal fat is also a source of omega-3 fatty acids, EPA, DHA, and DPA. If we are eating grass-fed meat, there will be some fat with the cuts like ribeye and New York strip, but it's not a ton. Most of the fat is now trimmed off of our meat by butchers, so we have to specifically ask for trimmings or for the fat around the kidneys, which is known as suet. Grain-fed meat is fattier than grass-fed meat, but I have concerns about the accumulation of more toxins. Just like other minerals, including sodium, potassium, and magnesium, we can't make calcium and must obtain it from our diet. Some people fear that eating calcium-rich foods will increase their risk of arterial calcification, but when our diet contains robust amounts of vitamin K2, this won't be the case. Aside from dairy, which we'll discuss below, sources of calcium on a carnivore diet include bone broth, bone marrow, bone meal, and eggshells. Eggshells should be boiled prior to consumption to avoid any possible contamination with salmonella or campylobacter that may be on the outside of the shell. In order to maximize the amount of calcium contained within it, bone broth can be cooked in an acidic solution, usually with vinegar, for 12 to 24 hours. Most of us don't appear to be well adapted to dairy from ruminant animals due to a number of proteins contained within it that can activate our immune system. The most common of these is beta-casomorphin, a breakdown product of casein. The name of this molecule looks like morphine, and it acts in a similar way in the human body. It activates opioid signaling pathways and can disturb normal satiety signals, leading to increased hunger and weight gain in many individuals. Exclusion of dairy in the clients I work with consistently allows for increased satiety, less inflammation, and easier weight loss. If we have an autoimmune issue or are interested in losing weight, I'd recommend leaving dairy out for at least the first 60 to 90 days of a carnivore diet. This includes milk, yogurt, butter, and ghee from all animals. Boron appears to play key roles in bone mineralization and hormone synthesis. Interventional studies with this mineral in both men and women have shown improved levels of testosterone and other hormones, as well as improved outcomes in people with kidney stones and arthritis. Tier 5 is basically how I eat day in and day out. As I've noted earlier, this type of a carnivore diet may not be for everyone at all times, and I don't think that everyone needs to eat just like me in order to derive the benefits of a carnivore diet. Traveling makes eating high-quality animal meats, organs, and fat difficult from time to time. The equation for living a radical life is simple. Eating animals nose to tail provides all of the nutrients we need to thrive with no associated toxins. We must also be outside in the sun, playing, hunting, and exploring, and sleeping in the dark at night, free from the incessant low-level stress we all face in Western societies. A bit of heat, cold, and some intense exercise are other important parts of this equation, as is spending time with family and community while giving and receiving love as we pursue something we are passionate about in our lives. The half-life of the main type of antibody, immunoglobulin G, used in our body is 21 days. This means that when our immune cells react to an invader or a foreign molecule from food, it's going to take 90 days, or nearly 5 half-lives, for the antibodies produced during this process to vanish from our circulation. 90 days! This is why it's so difficult to understand which foods may be triggering our immune system and what can be so powerful about elimination diets. Within the context of a clean carnivore reset, the carnivore diet can also serve as the ultimate elimination diet. It can help us get back to a place of radically improved health from which we can add less toxic plant foods back into our diet with careful attention to possible recurrence of symptoms. It's not surprising that a carnivore diet, which is ketogenic in nature, has also been reported in the medical literature demonstrating efficacy in treating patients with type 2 diabetes. Testimonials. I know the lack of fat and meat in my meals always had me craving for more. I kept myself full with bottomless cups of green tea, coffee, and fibrous vegetables. Stomach distension was the only way I knew how to feel full. Full, yes, but never satisfied. It wasn't until I saw some of my closest friends transitioning their diet and experiencing immense relief and healing that I had even considered the possibility of veganism playing a role in my health deteriorating. I was struggling with digestion, brain fog, memory loss, fatigue, and lack of sex drive, which were all deeply affecting the quality of my day-to-day life. I realized that the only time I was entirely free of digestive discomfort during the extent of my vegan journey was when I was barely consuming enough calories. Hence, my digestive system didn't really need to work very hard. My brain fog and fatigue were steadily increasing. My ability to concentrate and communicate clearly was practically non-existent. My first non-vegan meal in almost 5 years was 8 ounces of smoked salmon. I don't think I will ever forget the way I felt waking up in the next morning. My mind felt more clear and focused than it had been in years, and for me that was enough. My illusion of veganism being the superhuman diet was completely shattered overnight. I was humbled, to say the least. If the idea of removing plants from your diet long term is overwhelming, just think about it as a 45 or 90-day experiment and do the clean carnivore reset. I am confident that at the end of this time period, you will be amazed by the positive changes that have occurred. Common pitfalls when starting a carnivore diet. One of the most common pitfalls explorers of the carnivore lifestyle encounter is what we might call disaster pants, aka loose stool or diarrhea. It's certainly no fun, but it usually only lasts for a few days. In some people with underlying gut issues and lots of nasty microbes previously ruling the roost, loose stool can last up to a few weeks. Diarrhea at the beginning of a transition to a carnivore diet is often the result of shifting populations of bacteria within the gut, as the carbohydrate-loving bacteria die off. It can also be the result of increased bile acid secretion by the gallbladder and biliary tree, which are needed to emulsify the fat content of the diet. Normally, bile acids are reabsorbed in the small intestine, but with these increased amounts of bile, the small intestine may need some time to adapt. As a result, non-absorbed bile acids, which can end up in the colon, can often cause diarrhea. Loose stool isn't a reason to stop eating a carnivore diet. When transitioning from a plant fiber-heavy diet to one that is composed of mostly animal foods, it's completely normal to observe changes in stool quality, frequency, and volume. Specifically, those beginning a carnivore diet usually experience decreased frequency and volume of stool, with significant improvement in quality and resolution of gas, bloating, and pain symptoms. There are thousands of people eating a fully carnivorous diet who have easy-to-pass bowel movements on a daily basis. It is clear that we don't need plant fiber to go number 2, but some do report constipation when they begin an animal-based diet. Often these are people who had constipation prior to starting a carnivore diet, and who then observe an increase in the amount of time between stools when they transition to more animal foods. During the first 3 to 4 days of the transition period, we may feel tired, irritable, and achy. Symptoms sometimes are referred to as the keto flu. There is no infection here, however. It's our body gearing up the metabolic machinery to burn fat for fuel. Though sodium has been vilified for years by the mainstream establishment, it is critical to our survival and serves many vital roles in the human body. Its balance is tightly regulated within our physiology, and when levels are low, we waste other important minerals like magnesium and potassium in our urine. If we don't provide enough sodium to meet the increasing needs at the beginning of ketosis, our body may become depleted of not only sodium, but magnesium and potassium as well. Many of the symptoms of the keto flu are likely due to the electrolyte imbalance and can be ameliorated with increased attention to intake of sodium, magnesium, and potassium during this transitional period. Though eating steak for every meal is probably the easiest way to do a carnivore diet, for most people this isn't going to provide enough fat. In extreme cases of lean meat consumption, the result may be a condition that is referred to as rabid starvation that results in excess ammonia production. We definitely do not want to construct a carnivore diet out of just lean chicken breast, lean hamburger, or lean steak. This is a recipe for failure. Losing weight is about creating a caloric deficit, and the easiest way to do this is to eat satiety-promoting foods. Of these, good quality animal fats are king. In the Western world, we aren't used to eating trimmings, bone marrow, and suet, or thinking of these foods as valuable, but they are hidden gold. Grass-fed fat contains a number of valuable micronutrients, including vitamin K2, omega-3 fatty acids, EPA, DHA, DPA, vitamin E, and conjugated linoleic acid, CLA. It's also just darn delicious. Another reason for fatigue when transitioning to a carnivore diet may be an inadequate sodium consumption. How much salt should we be eating per day? Depending on body size, most people find 6 to 10 grams, 1.5 to 2 teaspoons of salt daily to be ideal, though some prefer even more. Though most people have improvements in sleep, occasionally people do report insomnia on a carnivore diet. In these cases, I again think about too much protein, too little salt, and disordered methylation related to not enough organ meats. We need a number of nutrients for the methylation cycle to work properly. The two most likely to be deficient are riboflavin and folate. Muscle meat just won't have enough of these for most people, but adding liver and kidney to the diet will improve things significantly. Early on in the adaptation period, some people do experience muscle cramping with ketogenic and carnivore diets. This is likely due to the electrolyte deficiencies of sodium, magnesium, and potassium, with sodium being the most common deficiency and the most important to correct first. If we are getting 6 to 10 grams of a mineral-rich salt and are still experiencing muscle cramps, it might be worth considering magnesium supplementation. We should also look at our diet and be sure that we are getting ample amounts of calcium and boron. If muscle cramping persists after addressing sodium, magnesium, boron, and calcium, I would recommend working with a physician to look at the levels of a wider array of micronutrients. White spots on the fingernails are one of the clinical signs I often see that indicate a nutrient deficiency. This is a non-specific finding, however, and can be related to a lack of many different nutrients, including zinc, calcium, selenium, manganese, copper, and other minerals. If we're not digesting animal meat or fat well, it's likely because of a nutrient deficiency that we've acquired by not eating these foods rather than an indication of an intolerance towards them. To digest meat, we need a strongly acidic environment within our stomach. In order to generate all of this acid, cells there must have enough zinc and other nutrients commonly found in meat. Zinc supplementation has been shown to improve gastroesophageal reflux by increasing acid production and leading to better functioning of the gastroesophageal sphincter. Guess how many sources of zinc there are in the plant kingdom? 0. What's a good source of zinc in the animal world? Red meat. Bile salts emulsify the fat within this partially digested food, allowing it to become water-soluble and creating a greater surface area for pancreatic enzymes to act upon. If we don't produce enough bile, fat in the food we eat will pass through our gastrointestinal tract and be excreted in a condition known as malabsorption. The result will be a pale-colored stool rather than a stool with a normal brown hue. If a person has trouble digesting fat and has pale stool, it may be the result of inadequate bile production. This condition is usually due to a choline deficiency, which has been shown to impair proper transport of bile acids into the bile from the liver. Bile acids are synthesized in the liver from cholesterol and then transported into the bile ducts, which flow into the gallbladder in a choline-dependent manner. If we don't make enough bile salts, or the bile salts made in the liver can't be transported into the bile, the cholesterol concentration in bile can become too high, leading to the formation of the most common type of gallstones. If we have gallstones and still have our gallbladder, or if we want to prevent gallstones, we should be sure to get a robust amount of choline in our diet, egg yolks and liver for the win. Animal foods are what our body really craves. They provide ultimate nutrition without the toxins found in plants. If we eat the same plant food day after day, the specific toxins contained within it build up in our system and we steadily develop aversions. Remember, plant foods are survival foods. They aren't meant to be eaten regularly. Once you begin a carnivore diet, I think you'll be surprised at the way you look forward to eating every meal. Animal foods never seem to get old, boring, or repetitive because they are what we are programmed to eat, and they are incredibly nutrient-rich. Eating a carnivore diet allows us to join the elite ranks of those with exquisite insulin sensitivity, and having an ApoE4 polymorphism won't be harmful in this situation. In fact, when used in individuals with ApoE4 variants and cognitive impairment, high saturated fat ketogenic diets lead to improvements in markers of insulin resistance as well as brain functions that include memory, executive function, and abstract thinking. The end of the road and beginning a new way of life. Beef as a whole represented only 1.9% of the total greenhouse gas emissions in 2016. 1.9%. That's half as much as plant agriculture, 10 times less than industry or transportation, and more than 15 times less than electricity generation, the largest producer of greenhouse gases. Methane emissions from livestock is part of the carbon cycle and does not increase the total amount of carbon in the atmosphere. In contrast, the carbon dioxide produced by industry, transportation, and electricity generation represents newly created carbon and adds to the overall amount of this element in the atmosphere. As part of the carbon cycle, methane produced by ruminants goes into the atmosphere and is broken down into carbon dioxide after a period of about 10 years. This atmospheric carbon dioxide is then used by plants during their cellular respiration to make carbohydrates. Cows then eat these plants and digest these carbohydrates, releasing methane back into the environment as the cycle continues. In place of ruminants, many suggest that we eat fake meats made from sawdust, bamboo, and oxidized seed oils. They state that such choices are better for the environment, ignoring the fact that these industrially produced burgers contribute more to greenhouse gas emissions than well-raised ruminants. Furthermore, the monocrop agriculture that is used to grow most plant foods today damages the soil and reduces organic matter by exposing deeper layers of earth to oxidation in the process of tilling. Once these layers are damaged, they are much more susceptible to erosion and runoff when rains arrive. If we really want to flourish and become the prodigious beings we are designed to be, we must remember more than just how our ancestors ate. We must remember the way they lived. The carnivore diet is about adopting a lifestyle in which we make efforts to do what we were designed for as often as possible. It's about walking in the woods with bare feet and taking the time to play. It's about moving and jumping outdoors, in the sunlight, in the snow, in the cold lakes and rivers. It's about celebrating the communities around us that give our life meaning. Let us not forget that one day we will all leave this earth. Perhaps nothing is more important than living the short number of days that we have here with as much joy and vigor as possible. Frequently asked questions. Grass-fed meat is better than grain-fed meat in so many ways, from the environmental perspective, the nutritional perspective, and the toxin perspective. Cows raised and finished on grass are superior sources of food for humans. Grass-fed meat is also a more nutritious food source. Studies of grass-fed meat have demonstrated higher levels of vitamin C, vitamin E, and glutathione when compared to traditional grain-fed meat. They also provide a more favorable fatty acid profile with increased levels of EPA, DHA, DPA, and CLA. Intuitively, this makes sense. Cows and buffalo fed the foods they've evolved eating are going to be much healthier than those confined to a feedlot that are fed inferior food. The most striking difference in the quality of grass-fed versus grain-fed meat are found when we compare the toxin loads between these two. Grain fed to cows is sprayed with pesticides and often highly contaminated with mold. Though meat cooked on a grill or at high temperatures tastes pretty good, there is some evidence that heterocyclic amines and polycyclic aromatic hydrocarbons do induce some degree of oxidative stress, activating the NRF2 pathway in the liver. Using low-temperature cooking methods like a pressure cooker, crock-pot, steam convection oven, or slow cooking in a pan will lower the formation of HCAs and PAH significantly. If we elect to cook our meat in a pan, I'd recommend against the use of oils, as these can become oxidized with heating. The best option for this is to use a high-quality stainless steel pan. Avoid using non-stick coated pans of any kind, as the chemical coatings on these can also be toxic for humans. I prefer actual fat from animals to the liquid rendered fats like tallow or ghee that are more common today. Unrendered fat may contain more fat-soluble nutrients and has accompanying connective tissues that provide a source of collagen. It's also easier to digest for many people. With our acidic stomachs and long, small intestines, we are uniquely adapted to absorb the nutrients present in animal foods very efficiently, but the same cannot be said for plant foods. The simple proof of this can be found in our poop. Those eating a carnivore diet quickly notice that the volume of stool decreases significantly, and undigested food is never seen in the toilet after bowel movement like it might be after eating salad or other plant foods. The notion that meat putrefies in the colon is just plain wrong as well. Unless there is a major problem with our digestion, undigested proteins from animal foods do not make it to the large bowel in any appreciable amounts. During the 8 to 12 week period of keto adaptation, our needs for sodium appear to increase significantly as insulin levels drop sharply. Many people find that during this period and beyond, intakes of salt as high as 6 to 10 grams per day can be very helpful. Large epidemiology studies suggest that those consuming this amount of salt have the lowest cardiovascular death rates and overall mortality. It seems likely that our ancestors would have sought out salt deposits to obtain this nutrient, as many other animals do. In more recent history, salt has been treasured and used as a form of payment, from which the term salary is derived. Spring water is much higher in magnesium and other minerals than the municipal tap water available today. The mineral water Gerolsteiner contains 100 milligrams of magnesium and 345 milligrams of calcium per liter, and tap water simply pales in comparison. Muscle meat also contains a decent amount of magnesium with about 100 milligrams per pound. Drinking a few liters of spring water per day and eating as little as 1 pound of muscle meat can easily get us to the RDA for magnesium from highly bioavailable sources. LDL probably rises on a carnivore diet due to increased production of cholesterol within the shared synthesis pathway with ketones. I don't think a rising LDL on a ketogenic or carnivore diet is anything to worry about as long as markers of insulin sensitivity and inflammation are low. We understand that organ meats may be very nutritious, but we haven't grown up eating them, don't know how to cook them, and find the flavors or textures to be odd. In this situation, desiccated organs from high-quality grass-fed animals are a great option, because the freeze-drying process is very gentle and involves low-temperature dehydration. It preserves the nutrients in organs very well. Let satiety be your guide. If you are hungry, eat. If you're not, don't. One of the challenges for many individuals as they transition into a carnivore diet is breaking old habits of snacking or using food as entertainment between meals. When transitioning from eating this way, many find that their cravings are significantly decreased and are much less tempted to cheat on a carnivore diet. I don't count calories, nor do I think you should need to on a carnivore diet. If weight loss is your goal, incorporating fasting into your carnivore diet occasionally or on a regular basis will help you achieve your goals sooner. For those of us at a healthy body weight but with autoimmune or inflammatory issues, fasting might also be a useful intervention because it allows the gut to rest and provides an opportunity for the immune system to calm down. Though fasting is touted for its autophagy, cellular housecleaning benefits, remember that a ketogenic diet leads to many of the same biochemical changes within the body, even in the fat state. Time-restricted eating will also result in some autophagy during the daily fasting window, an effect that will be accentuated by low-carbohydrate diets. A carnivore diet is not about limiting what we can eat. It's about understanding that on this type of diet, we are able to eat the most life-giving, nutrient-rich foods on the planet all of the time. It's a diet of abundance for us, as it was for our ancestors who treasured animal foods when they could obtain them. Using cheat meals incorrectly shifts us into a scarcity mindset and positions junk food as abundance. Ketogenic diets have been shown to decrease oxidative stress at the level of the mitochondria, to reduce blood pressure, and to be neuroprotective in models of traumatic brain injury. Multiple studies have found that after a short period of keto adaptation, low-carbohydrate diets do not increase the level of cortisol or activate the sympathetic nervous system. Ketogenic diets are very safe, and there's a substantial amount of evidence that they are protective in many ways. Studies of ketogenic diets in endurance athletes suggest a performance advantage with this way of eating and show that after a 6 to 8 week period of keto adaptation, rates of glycogen storage and replenishment are equivalent with those of carbohydrate-focused athletes. During very long aerobic exertion efforts like a marathon or beyond, we will need to refuel during races and in training in order to avoid complete depletion of glycogen stores. This is probably best done with a clean source of carbohydrates like dextrose or honey, rather than highly processed gels and other supplements. For efforts that aren't long and intense enough to deplete glycogen, carbohydrates are not necessary during workouts, and consumption of a carnivore diet after exertion will allow for full replenishment of glycogen stores. Whether or not we choose to use carbohydrates will depend on our sport of choice and goals, but I believe that most will not need them once they have allowed time for keto adaptation. If strength gains are our main goal, eating more frequently throughout the day to trigger mTOR as much as possible would be a reasonable strategy. The major pitfall for women appears to be inadvertent calorie restriction due to the satiating effects of these types of diets. If you are looking to lose weight, this is a good thing, but women looking to maintain weight or gain muscle should be careful to ensure adequate calories. In a caloric deficit, menstrual irregularities may occur, but with attention to this aspect, most women should not experience any irregularities with a ketogenic or carnivore diet. Being in a state of ketosis has been a common occurrence for pregnant mothers, infants, and young children throughout history. A nose-to-tail carnivore diet is rich in and provides all the nutrients we need to be fertile and to nurture healthy children. Getting enough calories to meet the increased requirements of pregnancy and lactation will be key if we choose to eat a carnivore diet during these periods.